Great. Lo streaming è partito?
Sì, sto guardando.
Ok.
Vediamo ancora un secondo. Warning. Bitrate. Quindi, eccolo. Bene, siamo pronti.
Great. So welcome everybody for this new day of with many exciting lectures. So before we start, just a reminder of the few rules on how to ask questions. So if you're following from YouTube, you can ask questions in the chat and I read them for you. If you are connected to Zoom, you can either post them in the chat or use the raise hand feature of Zoom. Great. So it's my pleasure to introduce again Andrea Rinaldo, who is giving the second of 3 lectures. So thank you very much, Andrea, for being with us. Please, you can share the screen when you are ready.
Thank you so much, Jacopo. And let me go back to the screen. I assume that unless I hear you lamenting that you don't see, I would assume that my full screen will be visible to you all. So welcome back to my the second of 3 classes on river networks as ecological corridors. I will subtitle Species, Population, and Pathogens. You may remember, if you attended the previous one, that essentially the leitmotif of my talks is that we claim that dendritic substrates, like river network instead, with certain recurrent properties that we have analyzed, however briefly, but those dendritic substrates for ecological interactions bear important consequences for a number of processes that we'll be analyzing. That is essentially patterns of biodiversity, which I haven't done, I'll do today in part, the rivers, the spread of species along the network itself, so biological invasions in a sense, and the spread of waterborne disease, which we'll see in my last class with reference to epidemic cholera and endemic disease like proliferative kidney disease in fish. And the idea is that the system is so constrained by the substrate, the nature of the substrate, recurrent properties, local properties embedded in it, that in reality it would endow the system a certain degree of predictability beyond what was originally thought. And so what we have seen during the first class was that once you put those constraints in place and you use even the simplest possible model, in that case was a neutral model of biodiversity, whether you're using a meta-population model or a meta-community model, whether you're using nearest neighbor interactions, or you're taking a kernel, which goes from a certain discrete length, or a mean field approach in which you average over the whole ensemble for getting replacements, or randomly selected species removed from a single site, the system tends to have certain indication. That is, the very nature of a substrate of interactions with the dendritic network has important consequences, which we have later verified by a number of different tools. So the main argument that I put together, and it's a one that said that what these lectures try to do is to draw together several lines of arguments to suggest that an integrated eco-hydrology framework that blends laboratory, field, and theoretical evidence has contributed substantially to our understanding of the form and function of the river network. What I shall do today, I shall begin with talking about biological invasions, possibly biological invasions on fractal supports, like a tree, which is the ones that we have seen in the case of river networks. So what you see in here is a map of invasion of the zebra mussel along the Mississippi-Missouri River system in the northern United States. And you see different coded by different colors what happened to a species that was completely alien and introduced by accident into the system. We're talking about hydrochloride, and to make a first example, we're talking about human range expansions and the population migrations in the past, which is an interesting idea by the group of Spanish physicists, which I'll be putting forth today. So first, let me start with the standard things many of you have seen in a number of contexts, and it is the Fisher-Kolmogorov traveling wave approach to a system. So if you have a model in which you have dispersion rho, in this case, is like a density of a species, or you name it, of any kind, moving along a one-dimensional support. And mind you that in the case of river networks, makes absolutely good sense to think of a one-dimensional system in intrinsic coordinates, because the width and the depth of a river cross-section are much smaller than the longitudinal length you have to travel. So the assumption that making open channel flows and flows in pipe, etc., as a one-dimensional thing, it's absolutely a standard assumption verified by hundreds of years of practice. So essentially what we say is, suppose that the system would obey a simple diffusion with constant coefficient, so the term d square rho over dx square, coupled to a significant over the time scale of diffusion reproduction system. That is, in this case, a logistic equation in which you essentially the reproduction of a density follows initial phase, which is the exponential growth phase, the Malthusian so-called dynamics, later on curbed by reaching an asymptote. So the growth term is nil, either where the density is 0 or where rho reaches a carrying capacity K. Why this is interesting? This has been studied for a long time, and notably from the most gifted applied mathematician of the last century, Kolmogorov, in fact, and R.A. Fisher, the inventor of so many results that are crucial now to modern genetics and molecular medicine, etc. They studied, they gave under different angles, they saw the same result. What happens if you suppose that, for instance, you start generating at a single point in space over a length L, and you inject a single shot of a certain number of organisms of the species whose density we're planning to analyze. The system here is known to generate a soliton, that is an undeformed wave after a short while, that propagates without the constant celerity, if it obeys this equation, which is solely dictated by the value of the growth rate inverse of time and the diffusion coefficient length squared divided by time to generate a characteristic velocity, which is twice the square root of a times d. How do you prove this result? Well, it's an interesting point, which you essentially take the diffusion equation under this limiting term, and you do that initially by assuming that this term is negligible. And what you have is that you can calculate the expansion radius of the most of a mass, or a assigned fraction of a mass, which you have in your system, and how this expands in time. Thereby, what you're studying is the behavior, and that has an important feature for what concerns our capability to describe the network effect. What happens in the vicinity of a low density when this starts propagating, actually. So the idea is that if you calculate the expansion radius, that in a regime in which this log of a quantity, whatever you want to have it, pales compared to the term in which you have the square dependence of the square root of a radius. So the velocity of a speed is essentially twice d times r, which is the growth rate in the Malthusian model, in which you eliminate the carrying capacity effect. And by no surprise, the carrying capacity, even if you extend this, it can be shown that the front speed is essentially, in the case of the Malthusian dynamic, still what happens in the vicinity of this reaction term for which the density approaches 0. And that's not by chance, because what we are saying is that, so essentially the same result of the Malthusian population, which if you take a proper computation of the expanding radius for a Malthusian population through the curve here, and the actual solution of the system, which you see that quite soon the system behaves exactly in the same manner. So what is interesting is that in a two-dimensional system, for instance, or in an isotropic migration system, 1, 2, 3, n dimensions, whatever you have it, the system will have essentially that the speed of a traveling wave or the organism generated that will reach a plateau and stay there and propagate on the fog, it's proportional to these two coefficients, ignoring the effect of the carrying capacity, which is immaterial, but only depending on the diffusion coefficient and the rate of reproduction, the reproduction rate one at a time. This thereby, in an, say, Eulerian support, but is in 1-2 dimensions, etc. And this will have some importance in what follows. Now, what is interesting thereby is that what matters, and that is important for the particular substrate we are studying, which is a tree that is a unique path for any node to any other node. It's a directed graph in which there is a sense of direction which is embedded in the structure of the system of Lorentz diffusion. And so essentially you can map the evolution of a density of organisms in space and time as a rescale variable of an undeformed property, which is the typical structure of the characteristic curve which I have there. So the fact that what matters is what happens in the vicinity of a zero density is important whenever you have to look at systems like this one. This is Peano network, which I introduced during the last question there. Why am I using Peano? I'm using Peano because it's a deterministic fractal rediscovered Giuseppe Peano, the Italian mathematician, who wrote a famous paper over a curve that fills the entire plane, which was considered like a blunder by mathematicians of this area, and we're still studying it 100 years afterwards. Benoit Mandelbrot, the genius of Mandelbrot, rediscovered it to support his idea that that mountains are no cones, coastlines are not broken lines, nor traveling does travel in straight lines. The fact that after 2,000 years after Euclid, we started looking at the geometry of nature with different eyes, was embedded in a curve which is strictly deterministically self-similar, because it's created by a process where it's from the originator. You essentially do the system which have a broken down half of a segment here, adding it into a system of this kind, and that allows you to calculate exactly a number of properties. Why this is an interesting feature? Because if you look at topological properties, of course, if you look at that, you say this is not a river network. Of course, it's not. But it allows you to calculate certain quantities exactly. For instance, if you count the number of, suppose this is a directed graph, and you can count, and if you suppose you have a seed point like here, for instance, or in this case, it would be here, if you assume that the thickness of a line is proportional to the total contributing area. It's a tree, so in any place you can calculate the number of nodes you have upstream. And what is interesting is that you can calculate exactly the number of sites equally distant from the outlet, distance being calculated in what is called normally chemical distance, that is along the structure of a network itself, which is hydrologically is a very important quantity, because if you suppose that it rains a given quantity in every single node of that, the distance, if a system behaves in a dynamically constant way, will means the arrival time at the outlet. So essentially, the response of the system will be related to this quantity. And in the case of Peano, this is an interesting thing, because this is a binomial multiplicative process solved exactly. It's an exact multifractal that has been studied by many, including Mandelbrot itself. In this case, what is specific of that is that even if by looking at this, you know that this is not a river network. However, the topological properties, which are non-distinctive in many respects, almost statistically inevitable, the topological structure of this is exactly indistinguishable, like bifurcation numbers, lengths, etc., from the ones exhibited by river networks. So the message I'm conveying here is that in this case, you can study exactly the behavior of a traveling wave along this backbone of propagation, for instance, because you can calculate exactly the number of bifurcations that the system will experience in its traveling downstream. So if you solve this numerically, let me show it directly. What you see, it's a diffusion plus a reaction term, a logistic reaction term, happening in this sense, what you're seeing here. And this is how the system propagates. What is interesting is that if you take the longitudinal direction, what matters, for instance, from going from source to outlet, I put it for clarity horizontally, but it could be even any source term would have exactly the same behavior. What happens is that if you do this, so this is how the paw propagates into the system, depending on different features. In this case, you have like no drift. I'll later on examine the case with have a significant hydrodynamic drift. And what happens here is that you're going to have a system in which the jagged part here is due to the heterogeneity is generated by the structure itself, but it generates a traveling wave. And what is phenomenal is that a walker that is at site on the backbone can spurt a certain time on the secondary branch. If you go here and you get sidetracked in this direction, you spend some time around the system here before you can eventually plug back and continue your travel towards the outlet. So the main point is that if you go through a system which is one-dimensional, but with significant delays because of a side tracking of the structure, essentially produces a slowing of the speed of a traveling front. So if you're only interested in the behavior of a system along the backbone, you can interpret the secondary branches as essentially a producer of the delay between sides of a backbone. So delays could be chemical, physical, biological reactions, but generates essentially of lifetime distributions, because that's what I'm interested in, into your system. And you can do that by a technique called continuous time random walk. I'm just leaving a few slides in here, but you have the density of a point is something in which you take into account the probability that you have of staying. You have a distribution of the delay time in a node. And mind you, the delay time in a node at this point is the time you spend if you get kicked out in one of the side structures. How long would you spend in the side pockets before you jump into the mainstream? And that's a distribution of a lifetime in here. And then you have also a term which describes the jump at every side, which in this case we call it reactive random walk, because you assume that at every time step you have a, with probability 1/2, you go back and forth, you have this system, you have no bifurcation. Or if you have z nearest neighbors, you have a probability 1/z of jumping in one of the ones you have. So, a long story short, this can be treated by Laplace transforms. It's called Hamilton-Jacobi mechanism. And you can calculate, in fact, fairly well via Laplace transforms how the system behaves. So you can calculate the equivalent of Kolmogorov-Fisher system for the homogeneous n-dimensional, in particular one-dimensional system, the speed of propagation of a soliton will be generated by the diffusion plus a logistic reaction term in this particular case. And in the case of Peano network in particular, you can do that exactly. So long story short, if anybody's interested, I told you that in the book that Marino Gatto, Ignacio Rodriguez, and myself have just published, there is a complete derivation of that. That speed, in fact, turns out to be something which is, if you look at the size of a jump which you assume that the system would have in discrete space and continuous time would be delta squared divided by tau, the mean waiting time of a system. You can see that this is the equivalent of the speed of the other, and this is the equivalent of a coefficient, so it's twice the square root of r times or a times d, but divided by a coefficient which is essentially the convolution of the waiting times you have outside the backbone over which you propagate, which is larger than 1. So a theoretical prediction on an abstract model, again, on a particular structure is telling you that in principle, you should have, but in Peano you can calculate it exactly, and you can do it numerically in any other case. The heterogeneity generated by the bifurcation structure that you will face propagating in one direction slows the front. And indeed, that's what happens. So if you take a two-dimensional system, this is what you have in the two-dimensional system of this kind. Like this is you inject into a particular place, oops, and you see how this propagates in space isotropically, and the speed is twice the square root of a times d. And almost regardless of a particular network structure, for Peano you have an exact solution. For OCNs, which you have seen the other time, you have something of the same kind. You have systematically a slower speed of propagation. You may wonder, who cares? And to do that, I resort to something which I find absolutely fascinating. a group of Spaniard physicists wrote on theoretical population biology in 2006, that started our interest in the subject, studied a quantitative model for the American colonization of the race towards the West in the 19th century, as seen through the lenses of transport of a fractal network. So the idea is that the landscape heterogeneities and the need of water forced the settlers about fluvial courses. That is, any settler will move along a fractal, in fact, because we moved along a river and only crossing divides at small times when they crossed the divides like the Rockies or whatever. Why do they need the fluvial courses? Because they need the water resources, of course, for drinking, energy, transportation, whatever. And the idea that the exact reaction diffusion model seems like a sensible way about a river network, it's in which you would have that the population growth will be a logistic with a rate parameter A for population growth at a single node. What is remarkable, that the archives of the US Congress allows you to evaluate, in fact, how the colonization came about. And what this guy had is that you had the yearly value of a relative frequency of that is the distribution of the relative frequency of the distance crossed year by year by the colonizers in different directions. And thereby, one of the key parameters that you have to calculate that the system would have been the phi distribution of the jumps, and was actually given and didn't need to be calibrated at all. And what is totally remarkable, if you take that, so you had essentially the growth rate from the archives of the population growth, you have the distribution of the features of a random walk in space, the distribution of the jumps that the settlers would go about over in an isotropic march towards the west. And what you had the Fisher bridge, which is then you have the parameters twice the square root of A times T, will be of the order of 40 kilometers per year. What is totally remarkable is that if you take any simulation, any kind of assumptions, etc., you go immediately to speeds of the order of 14 kilometers per year. And mind you, that the actual observed speed of the expansion was of the order of 1.3, 13.5. So the idea is that the data suggest that indeed, even in this case, the very nature of the substrate, in this case for the population, dynamics, was the one dominated by the features of a river network itself, because of the constraints that we have imposed on the system. This can be, so this is again the picture, so the constraints that we have imposed on the backbone of propagation of the traveling waves, facing the bifurcation that I will want that you will see inevitably the topological properties, that is the properties of the bifurcations that you would face along your path on any particular backbone will be the same in this structure, which is statistically identical to a two-river network, but replicable, etc., like we have seen in the case of OCN, and the Peano network, in fact, behave very much in the same manner. So yet another indication that the substrate plays a major role in the ecology of a system. So like I did, like I showed you the other time, the first thing that we did is say, okay, it's a strong suggestion, and can we study in the laboratory what happens? So we had developed the ability in my lab to work with protists, and so you have these essentially protistodromes, if you want. So we made these running places, and we see how we can measure it by more or less progressively more refined tools, etc., how an injected population in one end can invade at the speed at which it invades the system. So this looks like a Kandinsky picture, but is essentially the trace taken with microscopy done in my lab of individual trajectories of every single protist moving in this direction. And mind you that one of the two dimensions were much smaller than the longitudinal one, thereby making the one-dimensional assumption reasonable. So I am interested in how heterogeneity slows the front, and one would be to test how the demographic stochasticity pitches in with respect to the Fisher-Kolmogorov model of invasion. So essentially what we took the model of this kind and we decoupled essentially space from the intrinsic noise which you have in the system. Mind you that different realizations of a protist run through the system are things of this kind. So replication was absolutely needed to see how the system behaves. But the point is that if you do this and you replicate these normally, which had a 95% range, you see that still in a context in which you have, there's nothing neutral in here, there's nothing but demographic stochasticity induced by the biology of the system, and still what is phenomenal is that the Fisher-Kolmogorov system proves able to reproduce with a remarkable accuracy what happens in the laboratory. So interestingly enough, we would see, okay, now, can we generate other kinds of heterogeneity in the system? And how can we do that? Well, one idea was to use phototaxis in biological invasions. And the idea is to use the generalized receptor law, because you have a system in which every node, essentially, you have reaction in a node and you have transport among nodes. So the idea is to use the phototaxis in a system of this kind. This is my the runner, the drome, the stadium for this protist to run, the injection point and the collection point. And what we have in a phototactic system would be to use LEDs to generate resources heterogeneously distributed in space, which hadn't been done before. And slowing invasion by heterogeneous environments is one of the tenets in which I've been trying to get you interested in and had already seen. So the idea is that, for instance, heterogeneity could be spatial heterogeneity, in which, for instance, you put some drift in a system, so you put a bias in the system, not simple diffusion, but you add advection, which makes it even more heterogeneous, of course, because advection in this case you assume constant through the system, and you have a probability in any node. What you have is a probability which is an even probability of splitting in the case of diffusion, and it's not even if you have drift of any kind. So essentially what you have in the system, if you be a parameter that unifies the drift, what you get is that this is the wavefront speed in one case. So essentially in the case in which you have like zero drift in the square root of a t, like you have it in the Kolmogorov system, up to a system which is completely dominated by advection, which is important because I'm interested in studying the persistence of species in a river network season ecological corridor. So drift may or may not be important depending on ecologically viable quantities, like the nature of the organisms we are discussing. So anyways, what we know is that the Hamilton-Jacobi method we have used and the numerical simulations match fairly well. And if you instead put drifts, you start having negative and positive Fisher waves. And you can have like approximations, which are important in this case, which I'll briefly talk about, the so-called telegraph equation. Peano then stays in the system with features that are calculated exactly. Of course, I won't pest you with the details. They are quite involved, but again, straightforward and analytically solvable. And again, significant because Peano topological is indistinguishable from a real linear network. Although if you look carefully, you see that other metrics simply do not match. The telegraph equation is also something interesting. Of course, now, for instance, this is what happens if you look at how the system propagates, having chosen 3 different spots for colonizing the system. So this is how, in fact, the system propagation behaves. And there's a certain degree of commonality. There's a degree of commonality in the system here. Now, why the reaction telegraph model against the diffusion? Well, it so happens if you put a drift, what happens is that you have to assume that if you use a reaction random walk model, you essentially, unless you do some provisions for it, you would assume that the speed of propagation of the jump would be at infinite velocity, which is, in fact, it's a complicated thing to digest. Long story short, if you put a proper model which overcomes the inconsistency, well-known inconsistencies of a diffusion model in the case of bias, in this case, long story short, is that computational, nothing happens. So the reaction random walk describe what happens in the system, even in the case of drift, fairly well. So back to the system in which heterogeneity is imposed by me by choosing a certain distribution of resources with a certain correlation length. For instance, you have like silence, resources, silence. You see, it's a Poisson process, the one we modeled it with in the system here. So we had the ports at the bottom of a channel for changing the distance from the LED, the angle of light propagation and the like, to use something which is called the Keller-Siegel framework for calculating how the resource acts like some sort of a advection in the system. And I won't get into details. Of course, if you're interested, I'd be delighted to see what happens. But what is interesting in this case is that we do have a framework. So you turn off a light, the system behaves, and that's very clear from the spectral behavior of the system, behaves like a linear e to the minus the wave number squared times dt. So, so it turns out to be a normal diffusion if you turn off a light, and it comes up into something different if you generate a light field which aggregates and acts as a drift in the system, thereby disturbing the overall behavior of the system. So individual trajectories here are much larger. And as you see in the different realizations and different kinds, and yet a coherent picture appears. So this is light intensity profile we use in the experiment. And you see the one spread experiment was done for each landscape, and thereby you have a significant way of studying the system. What is the punchline of this relevant to our case is that the mean front propagation that we can compute theoretically through the Keller-Siegel framework and the experimental one, in fact, they show that the speed has a new actor. It is the autocorrelation length of the resource field. So heterogeneity appears in the system and affects the propagation of the speed or propagation of a traveling wave. And this autocorrelation does slow the speed, as you see here. In the case you're having there is where you have a mean invasion speed computed directly without any light field included, the square root of 2D system of that, which is the slope of the system here. So heterogeneity, whether given intrinsic to the structure of the river network, whose architecture and recurring characters we're long studying, and which I have introduced in my first class, have an effect. And to jump into something connected, like believe it or not, I'm going to a giant of a field, the late Ilka Hansky, Crafoord Prize a few years back, died too early, and whom we miss sorely, in fact, had invented a very important quantity that was key to metapopulation analysis called the metapopulation capacity of a fragmented landscape. That is, he found out that an ecological measure, super sound and quite well established experimentally from field work and from theoretical work, that characterizes the suitability of a substrate to effect of heterogeneity. In his case, was the fragmentation of a landscape, and there was the main motivation from the ecological viewpoint, of course. So technically, metapopulation capacity is the leading eigenvalue of an appropriate landscape matrix, which I'll explain in a minute. And essentially, the punchline was that a species is predicted to persist in a landscape, which is the matrix for ecological interaction. In my case, I would put this in the other, in the case of river network, is larger than some threshold value, which is driven by the properties of a species, and which is essentially the capability to disperse and the capability the dispersion capability and the whatever the mortality and reproduction rate. So metapopulation capacity is a number that can be conveniently be used to rank landscapes or for ecological interactions in terms of their capacity to support metapopulations. Metapopulation, of course, is another one of those bold statements, not unlike the neutral model of biodiversity, in that metapopulation essentially ignore interspecific interactions among species. That means there's no predator-prey relations, etc. So essentially, it is the intrinsic capacity of one species that determines its ability to survive. On this, I shall not spend issues, time, because of the limited time I have, but we have discussed that at length for the implications. So what happens is that the key to that is essentially a focus on the probability to be at point I in space, in time, of a focus species, the one whose ability to survive we are studying, which is the probability of being present in patch I at time t, which is a balance of colonization and extinction forces, and we just kill out the interspecific interactions over which ecologists spent lifetimes in the past. That was the greatness of one of the most, in fact, gifted field ecologists of our times, Ilka Hansky himself. So the point is the following: the rate of change of this probability in time is essentially a balance of the probability of being colonized by different places at distance, places site j, at distance dij, measured along the system, and that multiplies the colonization rate, and then you have the extinction rate, which is birth minus death ratios in the two. So the ratio E over C is what determines the feature that you have in there. Now, the main result is that what is lambda m, the eigenvalue of the system? Well, it's a landscape, it's the maximum eigenvalue of a matrix, which is a landscape matrix, which is essentially, this is the dispersal ability of a species, and this is the distance which you have in the system itself. So it's a positive matrix, it's a reducible matrix, in fact, so Perron-Frobenius theorem applies, in fact. And that's what he found out. And if it turns out that if the maximum eigenvalue of this particular landscape matrix, which is very easy to calculate for a landscape like ours, like I show in a minute, the landscape matrix for us, it's easier and more constraining than in, say, a savanna ecosystem like we had seen the other day. So what happens is the following: if you take a path along which an open channel network develops, so we started from any particular system, and we essentially streamline and simplify a network by assuming that the system would tends to minimize its energy dissipation, approximating the structures that and the statistics that a river network inevitably has. Well, like it or not, it's some sort of an unintended consequences. With a decreasing energy, the streamlining of energy, you have as a bonus, in fact, the better viability in abstract terms generated by the river network. So the ecosystem, in a sense, benefits from the physical process that determines the substrate that we are talking about. And then, well, there are other issues, but I realize that I've been chatting too much, so I'll be skipping fields. But anyways, this is a significant result in terms of the probability. If you take several realizations of the same network, etc., you have a band, an error band. And if you have like different kind of mean field versus OCN thing, you see that this is a significant result from a statistical viewpoint. So the last 10 minutes of my talk will be devoted to, can we use the same concept to study river networks and biodiversity? And I will leave to the third and last class how this, in fact, is key to evaluate how pathogens spread along river networks as ecological corridors, thereby propagating deadly disease, deadly or chronic disease. So what am I saying is that a river network is a set of, an oriented graph, my nodes and edges. Edges are physical edges obtained offline by digital terrain maps, as we've seen last time. You do have reactions, which will be physical, chemical, or biological reactions, in this case, biological, ecological reactions. And essentially, the links are what act as transport models between nodes. And this applies to individuals, this applies to species, or to populations. So essentially, we can talk about, rather than meta-population, meta-communities. And the first example I want to show is a paper which we care, Marino and I care very much for. Lorenzo Mar is the first author, on the meta-population persistence in river networks. What happens is the following. So the to study metapopulation dynamics can be done by looking at a system in which you may have like species that live on a substrate of this kind, which eventually might, in fact, get off and move off of a network itself. So the species, we are talking about the metapopulation, and that somewhat foresees what's going to happen with more complex compartmental models for a number of different phenomena. It would be like, you have like, in every node, you have two ecologically distinct developmental stages. So we split the population living in the node, which is a reach, if you please, in the young, non-reproductive individuals, Y, and the adult reproductive individuals. Movement from one node to the other can occur through different pathways, but along the structure of a network, either along the same network, if you are a fish, or even overland if you are an amphibian, for instance, in certain stages of your life. You take into account local demographic processes, birth, growth, and death, and the dispersal dynamics in each node of a river network, which again, using the full constraining power of a system of this kind. Now, this is important to explain the so-called the drift paradox, because to explain the long-term dispersal in system in which the drift could be very significant, in fact, is something which ecologically puzzled ecologists for a long time. For instance, to explain the long-term persistence of populations, for instance, empirically documented examples came in, for instance, for Scandinavian freshwater ecosystems, insect species compensate the larval drift that is transported passively along the river network with upstream directed flight of adults prior to oviposition. And it's called the Müller's colonization cycle. Otherwise, an excess production hypothesis has been forth in which drifting organisms would be exceeding the balance of demography of a local scale, which implicitly assume that drift essentially represents an extra mortality term for these kinds. But anyways, all I'm saying is that what is new here is not the ecology, which is well known, but it's the fact that you're using a substrate of this kind. And to give you an example of how coupled ODE is, in this case 2, for every node, at node I, you have the number of young non-reproductive individuals and the number of adult reproductive individuals. They have a certain mortality term, possibly density dependent. You have and what you have, most important, you have the probability of dispersing from any J site. So you may have adults at site I can be taken because of a connectivity matrix for any site J by simple proportion and transport that you have in the system. Whereas in the case of the adults, in addition to that, you have reproduction terms. It's not important actually how you do that, or the specifics of that, which is very well known system that we have in this place here. But what I'm showing you is the fact that how do you study those systems is 2 equations per n nodes of a river network. So you can actually assume that those matrices, dispersal matrices, are probability which depends on the connectivity structure, something which you assign and give offline, and which you can study by studying the stability of the metapopulation persistence is actually related to the stability of a population. That is, if a state x0 is stable, the population cannot persist in any of the river network nodes. If it is unstable, juvenile and adult abundances grow, and they grow the metapopulation persistence. So you can study the global equilibrium of those matrices, which is essentially, it's a super Jacobian in which you have 2n by 2n matrices that can be done exactly, and calculate the survival and the non-survival of the different ranges of the system. And for instance, one particular case which was particularly interesting was a study of salamanders, for which you had empirical data from, I think it was in Virginia, someplace, in which you can relate the conditions for metapopulation to persist and metapopulation not to persist, counting on the fact that the juvenile and the adults have different ability to jump off the river network itself. I am almost done, regretfully, so I will compensate next thing, but give me 3 minutes to complete with another example, the invasion of the zebra mussel, which paves the way of something which I'll be showing you Friday. That is, what you're seeing here, it was the zebra mussel was by accident introduced in the Mississippi-Missouri River Basin by introduction. It was taken from veligers, that are the larvae, were introduced in ballast water of cargo ships that came in from Europe, where it was native. And it started spreading through the system, reaching the DTAs, the embedded hydrologic units in the system have been growing dramatically over the years, so up to a point. But what is totally remarkable about this is the fact that at a certain point, it didn't even simply diffuse or disperse downstream. All of a sudden, you started having places where you started having flare-ups of invasions in completely unrelated places, far away from it. That introduces the fact that we can study river networks fairly well in those systems. And essentially, the local reaction equation is the local age growth model in 4 stages, which have larval production. Transport of larvae is a passive. Larvae are small. The veligers are essentially passive scalars, so they diffuse and they are vectored to the river network But most important, you have to introduce what is called modernly a multiplex network. Why? Because in those recreational ponds in which you put some boats, what happens is that if a guy picks up a boat, doesn't empty its ballast water properly, put it on the trailer and brings it thousands of kilometers away, then in as much as COVID-19 spread through human mobility, so did the spread of this system here. So the long story short, on which I shall stop in time, regretting that I have dedicated too much time to the early phases, but I will be saying this, what I miss from here next Friday. So here is the computed and simulated values in, like in this case, these are measured and these are computed, as you see in here, only by our ability to introduce those long-range flights generated in the injection by a different network, which is not related to the topology of a river network, but related to the road infrastructure and a certain probability of making a distance in the system, you are able to calculate one of the most devastating, because zebra mussel grew at a level of density that generated significant damage, not only ecological, because it displaced every other native species of that kind, but it also generated clogging of hydropower plants production and the likes. And this is an example of how, in fact, biological invasions reach the place. Yeah, this is a good point to stop in here. So the point line is that, and I will be taking on the next class to see how this, in fact, becomes the essence for studying through the same environmental matrix and possibly a multiplex network in which pathogens disperse, waterborne, I mean, through the network, and human mobility spreads the disease, how, in fact, you can use oriented graphs, nodal reactions, and hydrologic transport to tackle individuals, metapopulations, or metacommunities. Now, I'm ready for your questions.
Great. Thanks a lot, Andrea, for the very, very nice lecture. So we have time for questions. So please, if you want to ask one, you can use the raise hand feature, or you can type it in the chat. Yes, please, Monday.
Good afternoon from Nigeria. Can you hear me there?
Monday?
Can you hear me?
Yes, I do. Barely, but I can.
Yeah, okay. Yeah, can you hear me, sir?
I do.
Okay, okay. So I'm looking at the last point you presented in the slide. Is it, or do I join the class not to at the point you started? Is it that the network of the model that you brought was validated before you presented the result of the data, or is it a direct input without validation?
Well, it's a good question, actually, Molly. What happens is the following. Okay, there are networks and networks in this case. If you're talking about the transport of, you see the main point, right? You have nodes in which you have physical, chemical, biological reactions, right? And you have transport along the network structure. Now, if you're talking about the dispersal of the organism in the larval form, for instance, they are small seeds. They behave like a passive scalar. So they diffuse and they are advected and diffuse along the river network, right? But if you get a bunch of them and you take them from a node and you transport it completely artificially into another node, like you do with the alligators that get trapped in the ballast water uncleaned of a boat, then is the mechanism that can explain why you can actually have a flare up of a proliferation of a zebra mussel hundreds of kilometers away without any in between. Why this is interesting? Because it's like reseeding the infection, if you want, or the density of a population somewhere else. Now, in one case, the network is essentially the physical substrate, which we know directly, right? Because that's unavoidable. Digital terrain maps allow you to extract those connectivity matrices, that is how a node is connected to any other node in the system. And you can actually distinguish whether you, for instance, you can also you have a preferential transport downstream, but you may even go upstream with a probability which is different. So you have a biased transport. I'll be talking about that Friday. But in the case of a mobility network generated by the other, you have to find something else. So in that case, what we did was to evaluate a kernel of dispersion, but it was calibrated on the data actually, which is the displacement of commercial boats into the place through some proxy, which is a data on the number of boats parked in different positions on Mississippi, Missouri River Basin. So whereas in the case of a river network, it's a given, the connectivity matrix is given once and for all, because it's extracted objectively, it's extracted remotely and objectively manipulated. If you have a case, you may have to make assumptions. When we'll be talking about human mobility, for instance, to spread disease, which is not unlike these, you will have two possibilities. One, assume one of the models that are normally used, like gravity models, radiation models, you name it. Or you may use cellular phones and track individual mobility of large numbers. Now you can. Now, in that case, you may wonder whether the use of a telephone in certain places where cholera spreads is socially biased or not, if you're looking only at certain segments of a population. But mind you, my experience is that's not the case. But it's actually socially neutral, the ownership of a phone, even in the most dilapidated place on earth. And I've seen it with my eyes. And I'll tell you next week.
Okay, okay, sir. Thank you so much for the explanation. But I will appeal if there's maybe a kind of a reference material, then I would like to read more into it.
I'd be delighted. Well, actually, if I'm not selling anything because all the money will go to Cambridge University Press. But the book that I just published, that with Marino Gatto and Ignacio Rodriguez Iturbe, is called River Networks as Ecological Corridors, Species, Population, Pathogens, had everything that we have done the past 15 years on the subject. And a reference, hopefully, a good review of the literature as well. Thank you, Monday.
Great. Thanks a lot for the question and the answer. So we have time for more questions, if any. Also on YouTube, you can type the question if you want. Well, otherwise, I think we are actually on time. So otherwise, thanks again very much, Andrea, for the lectures, and looking forward for the next one.
Yeah.
Thanks a lot.
If I want to listen to Marino, can I stay on here?
Absolutely, yes.
Okay, perfect. Okay.
Yes. So what we're going to do now is that we are taking a break before Marino Gatto's last lecture, and we are going to be split in breakout rooms. So feel free to use them to chat freely with other participants. If you have a version of Zoom which is 5 or higher, you can switch room to chat with whoever you want. So we'll be back at 2:30 for Marino Gatto last lecture. Thank you very much. Great. I think we are almost ready to start again. So we have just to close the breakout rooms. And when people join, we'll start with Marino Gatto's last lecture. Let me see.
Eccoci, eravamo nella breakout room, siamo usciti adesso. Ho chiuso la breakout room. Mi senti?
Yes, yes. Grazie. Great, so...
Nessun altro computer? No, un attimo, no. Quando devo stare concentrato.
Great, so I think that... Great. I think that in a few seconds, people should join back to the main room, and we can start again. So just the usual announcements if you just joined us. So if you are following from YouTube, you can ask questions in the chat. I'm sure you have heard these many times. And if you are on Zoom, you can either use the raise hand button or ask a question in the chat and I read it for you. So I think that everyone is back in the main meeting room. So please, Marino, if you can share your screen, you can start. Thank you very much.
Okay, good afternoon to everybody. So let me share my screen and then optimize screen view. Okay, here it is. So, okay, so this is my third lectures. Okay, and the topic I'm going to speak today is unfortunately fashionable, and that is COVID-19. Again, I stress that in a way, my lectures and Professor Andrea Rinaldo's lectures are coordinated in a way. But COVID-19, however, is not included in the book that Andrea Rinaldo has been advertising, and I have also been advertising. Okay, so very briefly, the summary. First, I will talk about the context and the main epidemiological characteristics of COVID-19. Then I will talk about our model that we developed for Italy, which is a spatially explicit model. And then I will illustrate the model results up to the end of March 2020, which was the first month, well, the first 1 month and a half in Italy. And then I will show what might happen after the lockdown, might have happened after the lockdown. And I will talk about the scenarios and the possible containment measures, and then a few conclusions. Now, this is something I have already shown to you. I want to stress again that if you look at the statistics of communicable diseases, infectious diseases around the world, you see that the share of infectious diseases has been shrinking in a way along years. Of course, the number of deaths have been increasing because the population of the world has been increasing. But if you look at the share, the red share of communicable and also maternal, neonatal, and nutritional diseases, the share have been shrinking. And so around 2017 and probably the same for 2019, about 8 million. And now if you look at the statistics COVID-19, it is about 1.55 million deaths. You know that it is possible to linked to Johns Hopkins University site, and day by day, you see the sad statistic of global cases, which is probably a large underestimation of the real global cases. Global death is also possibly an underestimation, but not such a large underestimation at the global cases. And so we are now to a figure of more than 1.5 million deaths, which is really a very large share, and which means that COVID-19 is really a very important disease. In a way, it is not like other infectious diseases that might ravage the globe every year, like influenza. It is much more important. Now, let me stress that what I am going to present is actually a teamwork, a team of people with whom I've been working for a long time, and who are located in different universities and in different places. Although I must admit that they share one common feature: they are Italian anyway. Now, let me connect to the lecture by Professor Rinaldi on next Thursday, 11th, if I'm not wrong, to say that we have a lot of experience with spatio-temporal dynamics, and that is something that Professor Rinaldi already stressed in the past lecture. And so, for instance, he will show the progress of cholera and Haiti, and you see data on the left and model on the right, just to let you understand that there is a spatial signature in many cases. That is something that you should account for. And that is common to many diseases. So, for instance, the Spanish flu, probably you may remember that Spanish flu was probably the largest pandemic, I mean, with the exception of COVID-19. I mean, if we go back in time, the Spanish flu claimed more lives than the First World War. And actually, my grandfather, who was detained in a concentration camp in Austria, in Kathnau, because he was an Italian citizen, and unfortunately, he was in the Austrian-Hungarian Empire in 1915. So he was sent to concentration camp, but he died. He didn't die there because let's say, lack of food or anything. He died of Spanish flu. And if you look, for instance, at the spread of Spanish flu in the United States, you see how fast, how fast it was. So it started in the eastern coast, and then very rapidly, in a few weeks, it spread to the whole United States. Now, let me describe the characteristics of the COVID-19. First of all, the pathogen of COVID-19 is SARS-CoV-2. So, why 2? Because it is of the same family that actually caused the SARS. So it is an RNA virus. It has a crown-like appearance. It is actually due to spikes on the surface. So corona in Latin and in Italian is crown. So it's a beta coronavirus like SARS-CoV-1. And again, to stress the ecological importance of what I'm going to say, it is a zoonosis, and the host of several species of bats and rodents, in particular probably pangolin, maybe other rodents. And what are the main characteristics of the disease? Now, first of all, the main way the virus spreads is by respiratory droplets among people who are in close contact with each other. Now, let me show something. Now, this is an example of the aerosol emission when you breathe. Now, in this case, this is a study which was conducted together with a very famous orchestra. You know, I'm very fond of classical music. Sorry for that. So it is the Symphony Orchestra des Bayerischen Rundfunks. And here you see a way for visualizing the droplets and the aerosols that one can emit when singing or playing an instrument. Now, more rarely, you can get the virus via contaminated surfaces, but it is possible, so one should be careful about that. Now, another main characteristic that we know now, now let me provide a few technical terms which are shown here. So if you consider one individual who gets infected, he at first is not infectious. This is called the latent period. You may remember when we described microparasitic models that we were also talking about susceptible, exposed, infected, recovered, exposed, latent or exposed. This is the same term, which means the same thing, that you are infected but not infectious. Then at a certain point, you will become infectious. But that does not mean that you show symptoms. Symptoms actually, in general, show after a while. Now this period is called incubation, and then you become, symptomatic. Now, of course, when you're infectious, you can infect someone else. And again, the infectee will have a latent time, incubate the disease, become symptomatic, and at a certain point become infectious. Now, we call generation time the interval of time between the moment when the infector was infected and the moment when the infectee was infected. We call serial interval instead the time between when the infectee became infectious and when the infected becomes, the time when the infectee gets symptomatic and the time when the infected gets symptomatic, okay? This is called the serial interval. Now, the generation time, the average generation time, which under some independence hypotheses is equal to the serial interval, is about 6-7 days. So it means that the scale, the time scale for the infection is the order of 1 week. That is important. So from one infecting and one infected, okay, in the average, there's a week interval, 1 week interval, 6 days, 6-7 days. As probably all of you know, the asymptomatic fraction is quite high, larger than 50%. It may vary between countries. You probably know that it is more likely that old people show symptoms than young people. So the asymptomatic fraction might be higher in countries where there are a lot of young people, and let's say smaller in countries like Italy with a lot of old people. Now, one important message is that the maximum infectiousness occurs during the pre-symptom transmission. So that little time when you end to be latent and you are already infectious, but you've not developed symptoms. Okay, and that's clear for many studies. So it is about 5 days after being infected. Then what happens? That a pre-symptomatic, actually, it should be that we should call this period post-latent, because pre-symptomatics, some of them will never develop symptoms, so will remain asymptomatics, while some of them will become symptomatic. That's important for what I'm going to say later. Now, another thing is the mortality. Mortality is about 1–2%. Of course, it depends upon condition, upon the sanitary system, upon the fact that there are many old people, and of course, the old people have a higher mortality, and so on and so on. But we know now that more or less it's about 1–2%. And that might be compared, for instance, to mortality from influenza. It is something like 10 times larger. So there are two combinations that make COVID-19 in a way the perfect epidemic, the epidemic that unfortunately all the epidemiologists and the disease ecologists were waiting for. A large fraction of symptomatic people, and a mortality which is not so low as to make ourselves not very worried, let's say, but on the other hand, is not as high, like for instance, Ebola, to deplete the infected reservoir, the infected and infectious reservoir. Because you may remember that when I introduced even the simple SI model, I was stressing that the fraction of, that the prevalence of infected people and also the R0 were in a way decreasing with the mortality rate of the disease. So, and sort of the intermediate mortality, let's say, and a large fraction of asymptomatic make this pandemic so aggressive. And then, again, there is a spatial signature in this disease at the global level. So I'm going to show you this map and this video that shows how it all started, let's say, in China, and then it reached France and Germany, and then Italy, and then the whole Europe, and then, of course, started spreading to the Philippines and Korea, and then it reached the United States, and then it reached Canada, South America, Africa, Australia, and so on and so on. So again, it is clear that there is a very large, important part played by space, by how the disease develops in time and space. And therefore, it is very important to take into account space. And in fact, that was which in a way hit our interest, okay, because we have been studying many diseases in space and time. And when the disease reached Italy, and that was at the beginning of January, it is now clear that it was January and maybe even December. but that was not clear until the end of February. And if you look at, again, at the development, the spatial temporal development of the Italian epidemic, this is March 15, March 16, March 17, and so on and so on. So there is a clear spatial signature of this epidemic. And that's why we decided to work as soon as we can, very rapidly, on building a model for the spatial temporal spread of COVID-19 in Italy. And that actually what then materialized in this paper, which appeared the end of April, maybe on the Proceedings National Academy of Sciences, and that I'm going to illustrate to you. Now, first of all, the epidemiological compartments. Now, the subscript I indicates the nodes of the network that we're going to consider. And then we have the compartments of susceptible, the compartment of exposed, but then we had to introduce, with respect to the usual single compartment of infectious, 3 compartments, because we have the pre-symptomatic infectious, the symptomatic infectious, and the asymptomatic or mildly symptomatic infectious in each location I. And then, of course, if you are infectious and symptomatic, you might be hospitalized. Or if you're not too symptomatic, let's say you might be quarantined, or maybe you might die without even being hospitalized, or you are hospitalized and you might die, or you might recover. And if you're asymptomatic, usually you recover. Okay, so that's the basic, let's say, engine of our model with all these compartments. And here are the equations, and basically, again, the core of this local model in each node is actually the force of infection. Now, first of all, we can assume frequency-dependent contact rates. Now, it should be clear that in an unpopulated area, then you should assume density-dependent contact rate, but that's not the case of Italy. Wherever you are in Italy, basically, you are in populated areas, and therefore it is reasonable to think that every day you cannot have more than a certain number of contacts. So usually people have about, say, 10, 12 close contacts every day. So you can assume frequency-dependent contact rates. Now, so at the denominator, you have the sum of susceptibles, exposed, etc., etc. So the total number of individuals. And then at the numerator, you have the people who can infect. So the pre-symptomatic, the symptomatic infection, and the asymptomatic infectious, and they might have a different transmission rate. Because, for instance, we were expecting from other studies that the transmission rate of pre-symptomatic might be higher than those of asymptomatic and or symptomatic infections. So that's really the core. Now, if you go to the spatially explicit model now, you have the local model at each node, say the province of Milan, where I live, or the province of Padua, where Professor Rinaldo leaves. But then these nodes are connected. Of course, they are connected by mobility. There is mobility within each node, and there is mobility connecting the different nodes. So now the force of infection, when you go to the spatially explicit model, is much more complicated. In a way, you have to now introduce those mobility matrices I was talking about when I showed to you the model of schistosomiasis in Senegal. So the probability that individuals who are susceptible, pre-symptomatic, etc., move from site I to site J and contact individual people from site I who usually live in site I will contact individual in site J, and so on and so on. We even consider the probability that one individual living in site I and one individual living in site J will actually meet in site K, and one is infected, the other one is not infected, and so you have a close contact, and the one who is not infected becomes infected. Okay, and then of course we have the again this transmission rate, which depends on the stage, pre-symptomatic or symptomatic infectious or symptomatic infectious, and in principle might also depend on the site, because you might think that there are different behaviors in different sites and so on and so on. So this is the general structure of the spatially explicit model. Now, for this spatially explicit model, you can calculate that basic index, which is very much utilized and that I have introduced to you for very simple model, which is the generalized the reproduction number, which is, however, called now generalized because it is generalized to a model with a network. So again, let's consider the initial phase with no containment enforced. So we can calculate the basic reproduction number. And to do that, we can, as usual, start from the disease-free equilibrium in the spatial model, the no infection anywhere in all over Italy. And then we introduce a little bit of infection. Initially, the susceptible prevalence is 1. And then again, at R not equal to 1, we have a transcritical bifurcation. So you can run a bifurcation analysis on the model. And so you can find a transcritical bifurcation which occurs at R not equal to 1. Another equivalent way is to use the next generation matrix, which was introduced by Dickman, Esterbeck, and Matt. And one can show that R naught is the spectral radius of the next generation matrix. And in practice, this next generation matrix is actually built from the connection of the matrix CS representing the contact probabilities. And then you can recognize some specific times. 1 divided delta P is the residence time in the pre-symptomatic compartment. 1 divided by eta plus gamma A plus alpha is the residence in the symptomatic infectious. And 1 divided by gamma A is the residence time in the asymptomatic infectious. So in this case, because you can get infected from pre-symptomatic, from symptomatic or from asymptomatic, actually you have to sum these matrices, find the spectral radius of the sum of those matrices, and that will provide the generalized reproduction number. Actually, when you calculate, when you do the stability condition of the disease-free equilibrium and you go through the bifurcation analysis, you are considering the Jacobian of that complicated system at the disease-free equilibrium. And the dominant eigenvalue of this Jacobian is the initial exponential increase rate. On the other hand, elements of the next generation matrix describe the main routes of infection, and the dominant eigenvector of the Jacobian, which is the unstable manifold of the disease-free equilibrium, provides the geographic distribution, the initial geographic distribution of infections. And in fact, this is the way that we got this beautiful picture, and which is courtesy by by two of our co-authors, Enrico Bertuzzo and Stefano Micoli, which represent the main pathways, the main routes of infection in Italy at the beginning of the infection. And you can see Milano, Turin, Rome here. Okay. Now, of course, we had to calibrate the model. And here you see a logarithmic scale, semi-logarithmic scale, days, and so first patient in Codogno in February 19, and then the development of the disease up to the end of March. And these are the data that we've been using to calibrate the model. Now, the parameter estimation procedure, I want to devote, say, 1 or 2 minutes to that. First of all, when you have to calibrate the model, which is quite complicated, quite complex, there's always a trade-off between parsimony on the one hand and realism. So you cannot use too many parameters or too few parameters. So first of all, we had mobility from a national census at the municipal level, but we upscale mobility to the second administrative level, 107 provinces and metropolitan areas. The epidemiological parameters are not space dependent in this first paper that we wrote. The transmission parameters, beta pre-symptomatic, beta infectious symptomatic, and beta asymptomatics are not space dependent. And then, of course, we had to take into account that there were containment measures, so we should expect that these transmission rates would decrease. So we consider a sharp decrease within 2 days after the measurements announced on February 24 and March 8. And so we consider also the step reduction. Then, okay, we had red areas, and go into that. Of course, there was a reduced fraction of traveling people. How could we account for that? Through mobile applications, data that were collected by some colleagues from voluntary mobile data collection. And then there's an important thing, true spread of the disease from some initial foci, but there was not only one initial focus, although it was clear that the main infection foci were located in the northern part of Italy. But however, there were other foci. So we had to estimate also, in a way, the initial condition in the different in each province, and also the starting time of the epidemic. Also, we made another decision. The number of cases is not reliable. What is more reliable in terms of statistics is the number of hospitalized people, unfortunately, the number of deaths, and the number of patients discharged from the hospitals. So we used the Bayesian framework. We gave priors for the parameters. We sampled the posterior parameter distribution via the Markov chain Monte Carlo algorithm. And okay, technically, we use the likelihood according to negative binomial distribution. And Okay, sorry. And here are the results that I show for the whole Italy. These are the hospitalized people, these the number of deaths, but also for the hardest hit regions. Now, should be clear that these results are shown with reference to regions for convenience only, not because regions are isolated from one another. Because that's, you know, that's a common problem with many other problems, that they focus on the data of that region as if that region were disconnected from other regions. And the same is true for countries at the global level. But okay, that's another thing that we should discuss. And here you see a pictorial representation of the spread. These are the data. These are the calibrated model at the level of the second administrative level. And here is a fancy projection, you know, made by a simulation at the municipal level. But of course, okay, it is just in a way a fancy simulation. Now, one important thing of having a model that it is possible to make retrospective scenario. Because one question that we asked, were the containment measures effective? And the answer is yes. And you can estimate how effective they were by using the model. Because suppose, for instance, that no restriction had been taken. Okay, without any restriction, so no change of the transmission rates, no change in the mobility, and of course, no change in the people's behavior. That would have been the number of, no, sorry. Let me first describe hospitalized cases. That would have been the number of hospitalized cases. And then you can consider another scenario, February restriction, but no March restriction. And that is the second scenario. Now, this is the reality. So we had about 40,000 hospitalized people, a huge number, sufficient enough to send all our hospitals into big problems. But the number of averted hospitalized cases were about 180,000. So we would have had so much more without any containment measures. Another thing that you can estimate is the people who were infected, because the people that you measure, the number of cases that you saw in the Johns Hopkins site, of course, these are the cases that are discovered. People taking a swab and the swab being positive, or let's say now they're using this antigen test. Okay. Oh, I'm sorry. This is happening again. Okay, thank you. Okay. So you can estimate how many people were infected and possibly infectious at the end of March. Well, they were about 700,000, 10 times more than the official number of cases. But if the containment measures had not been taken, then the accumulated number of infected cases would have probably run to 6 million cases. So you see now that a model is really useful in this way, because you can also estimate variables that are not measured directly. So, main epidemiological result. We calculated basic reproduction number. It is about 3.60, well, very similar, 3. Let's say 3, 3.2, 2.8, something like that. That is a common number all over the world. We had a confirmation that the pre-symptomatic are extremely infectious because we estimated the beta, the transmission rate of the pre-symptomatic, and in order to fit the Italian data, that beta had to be much larger than the transmission rate of the asymptomatic or the symptomatic infectious. However, there was a large negative correlation between the fraction of asymptomatic cases and their transmission rate. Okay, so that's a problem. Containment measures had reduced the transmission rates by 45%, not enough at that time to make the reproduction numbers more than 1. That will be achieved later. And again, the model allowed the estimation of inapparent infections and the prevalence of susceptible prevalence of infected. Oh, I'm sorry. Then after that, we went on. Italy came out of the lockdown on May 3, so we started thinking, well, what is going to happen after May 3? Of course, everywhere, in Italy too, there was a concern about the economic consequences of enforcing a lockdown. And of course, the lockdown is a mess in the social and the economic terms. So we wanted to understand what might happen after the end of lockdown. Now, of course, the first thing we had to do, we had to recalibrate the model between March 25 and May 3, the end of the lockdown, which we did. And so we updated the model. And here again, you see at the end of April, beginning of May, the data, the cumulative hospitalization and the model. And then we also calculated the transmission reduction. The transmission was down to say 0.3, 0.4, depending on the regions and provinces. Okay, and in fact, you can see here, how about this again? I think you should stop annotate somewhere. Annotate. Let me stop annotate. Where is it? I don't know. Okay. Now what do I do?
I think if you have the annotation, you can do clear, and it should clear.
Because I don't see my mouse now.
Ah.
There's something wrong.
The slide looks nice though, so there is no sign or.
Okay. Now I felt that I can now move my mouse to indicate. Mouse. No. Well, anyway, let me go on anyway. So here you see the calibration, and then we try to have future scenarios. So first of all, we showed that there was a further decrease of transmission after March 25, transmission rate, fortunately. And then we tried to estimate some scenarios. So the blue line is the baseline scenario. Suppose that the transmission rate after the end of lockdown stays the same as it was during the lockdown. Or green and purple represent scenarios with transmission rates that increased by 20% and 40%. Now, why should they increase? Well, because, of course, mobility increases at the end of the lockdown. On the other hand, mobility increase might be effectively mitigated by personal protective equipment. Now people are free to go around, but they wear masks and so on and so on. And so let me show how effective that might be. So yeah, personal protective equipment. So let me show you.
verhindern.
Sicher singen geht, sagen die Forscher. 2,5 Meter Abstand zum Nachbarn nach vorne und 1,5 Meter zur Seite. Am besten mit Plexiglasscheibe dazwischen und ständig lüften. Masken sorgen zusätzlich für Sicherheit.
In this case, there's a surgical mask and a barrier. Okay. Then, of course, you might consider social and physical distancing. Then, again, you might implement again a lockdown at small and large spatial scale. And then one important containment measure, of course, testing, tracing, quarantine, hospitalization, isolation of people. And then, of course, you might have a combined implementation of all these containment measures. So, we sent the paper, we had reviews, and of course the reviewers, first of all, they say, well, you all know that review take time. And first of all, they say, oh, it might be nice if you could update the model. So to answer the reviewer, we updated the model up to June 15. And from here, you can see that Italy was actually quite, let me say, virtuous. That in a way, Italy, although the lockdown had been released, were following the blue line, so the baseline scenario, as if the transmission rate remained the same as they were during the lockdown. Well, with the exception maybe of Lombardy, the highest populated region in Italy, where, by the way, I happen to live. We also ran a sensitivity analysis to understand whether the fraction of asymptomatics, which is very unclear in a way, was, say, 10%, 25%, 50%, sorry, the fraction of symptomatic, in this case, 10%, 25%, and 50%, that is famous study in Voight-Gallion, blue. And it turned out that 25% is the most likely scenario, because that was confirmed by the estimated zero prevalence of the infected, a statistic which was available in Italy on July 15. And for instance, came out that Lombardia, 7.5% July 15 had been infected, with more or less, support the green line, the 25% that we were considering as a possible scenario. Of course, another containment measure that you can do is testing and tracing on a large scale. And again, what we did, we estimated for instance, the daily percentage of exposed and pre-symptomatic people that should be isolated. Now, it's very difficult to isolate exposed, of course, because in many cases, when you're exposed, the swab test is not yet positive. So it's probably more realistic to consider the infection generated by symptomatic cases and trying to trace, to test the symptomatic case, then look at the contacts of the symptomatic cases, positive symptomatic cases, and then look at the contacts, and so on. Okay, just to finish. One of the reviewers asked, are there possible scenarios after June 15? And okay, so, what about the problem? Suppose that there is immunity loss. So we ran scenarios when at September 1st, the transmission rate were back to the initial transmission rate, March, and with and without immunity loss. I would say that it is now clear that immunity lost, at least for a few months. Not clear yet, that there's not enough evidence, and hopefully we can hope that it is long. So these were the scenarios that we ran, let's say, without immunity loss, the one you see here for Italy, for Lombardy, and this is what is going on. We were assuming that there would have been a new lockdown on October 1st. Instead, the lockdown has been implemented 1 month later, and that is what is going on now in Italy. To conclude, so a spatial model, at least according to our opinion, including mobility, is fundamental if we want to project in real time the epidemic trajectories, at least at the very beginning, when it might be useful to implement immediately containment measures in order to stop the spread of the disease. In these ways, it is possible to estimate the demand for critical care, the hospitalization that you should expect in order to avoid the big problem for hospital, to estimate how much testing and tracing we should do. On the other hand, integrated model is also necessary. Age structure should be included, social contact structure should be included. Many other models that have been developed around the world, and even in Italy, also consider age structure and social contact structure. But unfortunately, I must say that they do not include space. Unfortunately, lack of available data is a problem. They are not made public. So in some cases, and in our case too, there was insufficient spatial granularity for some compartments. And if we look at the problem really at the global scale, then it is clear that there should be nested models from global, I mean, all the world, like for the problem of climate change, for instance, where you have global modeling and then nested, you have country models and then nested regional models, so that you can fine-grain the strategies, because the strategies might differ from location to location. And one thing that is unfortunately common to many countries around the world, that although the data were made available in a relatively short time, and you have seen the Johns Hopkins University site and so on, however, in Italy and in other countries, most of the data were not made available to the scientific community. The scientific community was requesting a wide availability of these data, but in many cases, this data, only I would say, a small part of these data, of course, anonymized data, were made available, and that is unfortunate. Well, I'm sorry it takes such a long time. I'm sorry, and I stop here.
No, I mean, it was very interesting. There is no reason to be sorry. Thank you very much, Marino, for the very nice lecture. So we have time for questions. So there are a few in the chat. So in one from Miguel Rodriguez, in the local model, is the parameter H, which I think represent the hospitalized individuals, capped by the maximum hospital capacity in each region, or is it assumed to be unlimited?
No, I don't remember. H, no, H, no, I don't remember what I indicated with H. So let me see. In the local model, let me see. Oh, the hospitalized, okay. The hospitalized people. No. Okay, the hospitalized people, or a very number, it is the number of people who are in the hospital at a given time.
Yes.
So,
Yes.
of course, it was capped by the hospital capacity by definition in a way, although, you know, the people were hospitalized and put everywhere. So it was really a mess. And it is now clear that at that time, many people were actually dying, even if they were not, without even being taken to critical care units.
Yeah, I guess this question alludes to the fact that the admission, for instance, to ICUs depends on the occupancy of ICUs, so the criteria change with the capacity. Then there is a question by Sylvia asking, what is the interpretation of the large negative correlation between the fraction of asymptomatic cases and their transmission rate?
Yes, okay. The interpretation is as follows, that if the number of symptomatic, if the asymptomatic case is large, very large, then the transmission rate can be in a way lower because it is the product, let's say, of the beta A and the transmission rate of A and the number of asymptomatic, because that will be, let me say, the viral load that goes into infecting the susceptible people. Now, you know, a good thing of Bayesian modeling, of Bayesian, statistical approach, is that you cannot only estimate the confidence of each parameter, but you can also look at the correlation matrix. And in fact, then you should, if you inspect the correlation matrix, then you find out that some parameters are not so much correlated with the other parameters, which is a good thing. It means that they are estimated, that they're well estimated. If you see negative or positive correlation, that means that the model is not so parsimonious in a way. And that's why we were concerned. And that's why we made a sensitivity analysis in the second paper with respect to the fraction of symptomatic and the fraction of asymptomatic people. And then the serological test confirmed that the fraction 25% of symptomatic and 75% of asymptomatic or mildly symptomatic, so that they don't even care, they don't go to the hospital, they don't take swab testing, and so on, is reasonable, at least for Italy.
Great. So there is a question about vaccines. Actually, given the good news.
Of course.
Yes. So a problem for 2021.
And we hope that we are doing that. And Ronaldo, I think, is still joining. Maybe no longer.
I am here. I'm here, Marino.
Oh, you're here there.
So the question, yeah. So the question is
I think that yes, we are working on that.
Yes. So can this model be used to optimize the spatial distribution of vaccination campaign, or do you think that it is of small importance relative to the demographic groups?
Well, of course, there are some things that pertain to the common sense, and actually these are the rules that in a way will be enforced by the European Union to distribute the vaccine. That you first vaccinate people in the hospitals, medical doctors, nurses, and so on, people who are at risk, people in, let's say, retirement houses, and so on. But then, of course, you can anyway optimize the vaccination campaign given some constraints. So the constraints of, say, the rules of the European Union, at least for Europe. Now the
Or the number of a batch of vaccines.
and of course the number of vaccines. Well, of course, that's
Well, the idea, if I may, may I, Jacopo?
oh, well, let's say 200 million for Europe, something like that.
No, but the question is whether, given a batch of vaccines and a set of rules, finding out the best distribution in space and time of that batch is still a thing which is precisely what we are working on now.
Yeah. And it's obviously not trivial. It's not trivial. Oh.
Great. So there is a question by Luca that says that, well, he really enjoyed your talk and he found very interesting the Bayesian approach. And he's asking, according to your model, which parameters are the ones that change the model results the most?
Ooh. Well, certainly, let's say the beta of the pre-symptomatic make a lot of difference. You know, I told you that there is evidence, that is experimental evidence, that the pre-symptomatic are very much infectious. And that's why we decided to include a compartment of pre-symptomatic. But it's not our invention. There were other people who had done that. But we found that that was fundamental. But then we didn't fix the transmission rate of the pre-symptomatic. And it naturally came out from calibration that the beta of the pre-symptomatic must be quite large compared to the beta of the asymptomatics and the infected symptomatics. And again, it came out of calibration that the other two betas, apart from that problem of the negative correlation of the beta of the asymptomatic, are similar in terms of the order of magnitude, which makes sense. Say, even from studies on the viral shedding. So viral shedding is actually decreasing after you get the symptoms. Or if you are asymptomatic and you are followed during the asymptomatic development of your disease, viral shedding is similar to that of symptomatic, but it is the pre-symptomatic with the highest viral shedding, more or less. Of course, then there are a lot of individual variations. I don't know whether I answered, Luca, whether I answered your question.
Let's see if answer in the chat. So is there any other question? Yes, okay, the question is answered. If not, well, next week we are going to have actually one of the roundtables. They should appear soon in the program if they are not yet appeared. One of the roundtables is going to be precisely about COVID and the pandemic, and Marino Gatto will be one of the panelists. So there will be more discussion about it and more possibility to interact about the
Yes, sure.
The most important topic of 2020. So thank you very much, Marino, for the lectures.
Thank you all. And okay, so I'll see you at the roundtable.
Yes. So thanks also, Andrea, for staying with us.
Thank you.
And so now we are going to break in the breakout rooms. Oh, sorry, there is actually a question that I didn't see. So I don't know if Andrea, if Marino is still here. Yes. Monday, please ask the question if you.
Yeah. Please, I am asking, can the generalized reproductive number have influence on the generation train of the model function Q? Is it possible?
Sorry, because I'm, you know, at my age, I cannot hear very well. Jacopo, you understood that better than me.
I think it's better if you type it because the communication is a little bit disturbed. I think you have a spotty line probably.
Okay, okay, I can type it right.
Yeah, thank you.
Okay.
Marina, I think Monday was asking about the generalized reproduction number applied to the COVID model.
Yes, but again, which kind of question?
I don't know.
You want to know more in mathematical terms?
No, I think it was a very precise question. I think he's typing it. So,
Is he using chat?
yes, I think he's typing.
Because I don't see
a spotty connection.
I was possibly writing. Okay.
Well, I can ask a question. Well, no, it's here. Please, can the generalized, okay. So if the generalized reproductive number have influence on the generational trend of the model function Q?
The model function Q. Now, what is Q? Wait a minute. Model function Q. Well, I'm sorry, but what is Q? The Q function? Because I don't see. Maybe I thought I didn't remember, but I don't see any Q function in my presentation. What do you mean by Q? Generation. Oh, quarantine. On the generational trend of the model function Q, the quarantine. So it's not function, you mean a compartment. So this, yeah, the quarantine compartment. Wow. No, it is just the opposite. That the rate at which you quarantine people influences the R naught, of course, because if you quarantine people, so there was, now let me share again my screen, maybe. Share my screen, okay. Okay, so there's you see a rate at which you quarantine people that are symptomatic infected, because at the beginning, at least in the first paper, at that time, Italy was not able to discover any asymptomatic. So it was a miracle if they did swab testing for the symptomatic. So you are symptomatic, and then you can be quarantined because the medical doctors might decide that you do not have enough symptoms to be hospitalized. And so you introduce a rate, and of course, the larger the rate, and the better it is in terms of decreasing the R naught, because these people are quarantined and they are no longer infectious. At least they are not so infectious. They're still infectious in the household, of course, unless they can isolate. They have a very large apartment, and in the family, you can isolate from one another, okay? But anyway, in any case, you are constrained to stay at home, and so you cannot go around and infect people. So the larger that, and the better in terms of the R naught. So, of course, if you quarantine a lot of people, that is a containment measure that will have a positive effect on the reproductive number. I hope that was clear. Okay, I see that you're doing so. Okay, so let me stop.
Yes.
Sharing.
Yeah.
Okay.
So thank you. Yeah.
Yes.
Is
fine.
Is satisfied. So thank you very much, Marino, again for answering to all the questions. So we'll take a 5-minute break before Jonathan Levin question. And if you're following from YouTube, the next lecture is not going to be live streamed, but we'll be back on YouTube for the lecture of Daniel Segre at 5 p.m. Italian time. So we're going to be split in breakout rooms. See you in 5 minutes. Thanks, Marino and Andrea again.
Ciao, ciao, Jacopo.
Bye all.
Okay, welcome back everybody. We are live again. This is Antonio Celani speaking, and it's my pleasure to chair the final part of today's session. And we have now the first of the 3 lectures by Daniel Segre from Boston University, and you can read the title by yourself, From Genome Scale to Ecosystem Level Modeling of Metabolism. Thank you, Daniel.
Thank you, Antonio. Welcome, everyone. It's a great pleasure to be talking to all of you. I'm talking from Boston, where it's mid-morning. I'm going to start by showing you the agenda. So we're going to talk today mostly about the logic of the cell, how to think about metabolism as a resource allocation problem. And we'll see next how to scale from genomes to ecosystems, in looking at spatiotemporal modeling and the long-term history of metabolism. So my hope for today is really to convey the way I think it's interesting to think about metabolism, and some, addressing some of these questions, hopefully motivating deeper insight. And I'll keep this fairly broad, so to make sure that everybody's on the same page. And we'll address questions such as, what is metabolism and why does it matter? Why should we be interested in studying metabolism? And why do we need mathematical models to understand it? And we'll see also, we'll start seeing why we can think of it as an ecosystem property. And I'll start by sharing something that I always find stunning, which is that microbes, despite being so small, can really change the destiny and fate of a planet. And in fact, they did. This is what you see here is the line of the amount of oxygen present in the atmosphere on our planet throughout the history of life, from about 3.8 billion years ago until today. And one thing that is clear here is that early on there was no oxygen in the atmosphere, barely any. And the reason oxygen started rising and becoming what it is today is due to the activity of bacteria such as this one, oxygenic cyanobacteria that photosynthesize, and in doing so produce oxygen. And it is because of these microbes that we have oxygen today, and it is because of these microbes that the planet changed completely, allowing the rise of multicellular complex systems, complex biological systems. So it's really important to understand how the metabolism of these organisms can affect at such a global scale other systems. Another reason to study metabolism, as of course all of you know, is that we are largely made of microbes. That is, many of the cells in our body are microbial cells. This is an estimate from Ron Milot and colleagues, showing that there is a little bit more bacterial cells inside and around us than there are our own human cells. So these are mostly in our gut, but there are microbes everywhere, and we're just starting to understand what role they play and how they interact with our body and with each other. And this also happens largely through metabolism, as we'll see. And there are many other reasons to be interested in metabolism, and by microbial metabolism in particular. Here are some examples. Some of you may be familiar with this, but there is a lot of interest in trying to understand how microbes can help produce biofuels and useful molecules from plant biomass, how they can affect global biogeochemical cycles, whether they can help plant and crop production. And there is a rising interest in how microbiomes in the built environment affect human life at different scales. Now, if you zoom inside and try to understand what is it that happens and that makes all of this processes happen, this is what it is. This is metabolism. Many of you have seen this chart hanging on the walls of biochemistry labs. In metabolism, what is one of the things that is beautiful about it is that really spans all scales of biology, from individual cells, again, to metabolism is happening in the biosphere. Each line in this graph is a chemical reaction. We'll see some examples soon. And this is a global biochemical chart that is the collection of all chemical reactions happening across all living systems. These are all now available on databases. But it's, one of the questions, how do we start understanding the history of this system and how it translates into all these processes that microbes are involved in. And I like to think of the hierarchy of biology in a way that maybe is a little bit unusual. We tend to think of how you go from molecular reactions, this would be a simple chemical reaction, and how this builds complex networks of, for example, the intracellular circuits in a cell, and this in turn leads to cellular dynamics, growth and division and so on, and then scaling up to ecosystem level processes. But what I hope we'll appreciate is that actually this process can go back and forth. And in fact, a lot of the modeling we'll discuss will have to do with starting from determining the molecular structure, but also looking at ecosystem dynamics and cellular processes, and trying to understand, based on what we know at the higher level, how those functions constrain the way molecular reactions should work. So somehow we can navigate this hierarchy back and forth, and not necessarily just building upward more and more complex systems. By the way, I should say, feel free to interrupt anytime. I don't know if I'll be able to see in the chat, but feel free to step in if you have any question or anything is unclear. I have an agenda for today, but if we don't cover everything, it's fine, and I'm rather be happy for everyone to have these concepts clear. So one of the questions we'll delve in between today and the next 2 hours is whether and how we can predict ecological interactions, that is, these green arrows, based on intracellular circuits, that is what happens inside each cell, and how do we do that? I will start by actually stepping back into something much simpler, what I like to think of as a sandbox for playing with toy metabolic networks. And this is, I think, a really useful exercise to start thinking about what is metabolism all about in a much simplified way, but it also raises a lot of interesting questions, as you'll see. So this is was motivated by work we did a few years ago with Sid Redner, Paul Krapiskin, Bill Reel, and it's inspired by other work on this string chemistries. And this is a artificial chemistry, a toy chemistry that is very simple. In this case, there are just monomers A that can be combined. For example, in this way, 1A plus 2A will give rise to 3As. You can express it in this way. And in general, you have this joining processes where a polymer of length I joins a polymer of length J to give rise to the combined polymer. And you can write the diagram for the complete chemistry, for example, all the way to polymers up to length 4. And you can see that this is fairly simple. And if you ask at this level, for this kind of chemistry, questions such as what is the most efficient way of producing A4 from A1, this is a pretty straightforward problem, which we can solve manually. And we want to have no waste of products and just use the minimal number of reactions. And the solution, of course, this logarithmic growth. From 2 A1s you produce A2, and from 2 A4 you produce from 2 A2s you produce A4. But this can get complex quite quickly. For example, if I show you the chemistry up to A7 and ask you how would you produce in an optimal way A7 from A4, this is something that would require a bit of thinking. You may have to break A4 into 2 A2s and so on and so forth, and gradually build up to A7. And I'm pointing you here, if anybody's interested in playing with this toy chemistry, you can create now, there is this tool we generated to produce arbitrary string chemistry with multiple monomers and different lengths, and you can play with this kind of scenarios. But I want to show you how this connects back to our real metabolism. And in order to do so, I will start by showing how one can find these optimal pathways for going from any initial molecule to any final molecule in this simplified chemistry. And just to give you an example, again, if you want to go from a molecule, again, of this string chemistry of length 1 to a molecule of length 6, you can make the 2 with 2 1s, produce 3, and so on. And this is easy, but there are some interesting patterns that emerge. For example, for some of these processes, the optimal solution, such as going from the 7 to 8, includes having cycles like this, where you need to have as an input a molecule that is generated by the process itself. So this is a little bit like an autocatalytic cycle where you need some of the internal molecules in order to bootstrap the activity of this cycle. And this is interestingly similar to a cycle that is present in real biochemical networks. And this is representing the carbon backbone of the TCA cycle, which we'll see shortly. It's a fundamental biochemical pathway present in almost all living systems. And it has this interesting property that is very similar. You have an input of a certain molecules. These are molecules with 2 carbons, but it needs the cyclic behavior in order to sustain itself. So this was interesting, but let's see, if we go back to real metabolism, why this is so important and how do we translate this analysis from our artificial chemistry to real chemistry. And of course, I don't have time, and I'm covering here in a superficial way material that could take whole courses, but I want to just give you an idea for those that haven't looked at the chemistry in recent times, what are the kind of molecules we think about and we'll take into account when looking at metabolism for microbes. This is methane, and it turns out there are microbes that can survive on methane as the only carbon source and the only energy source, which is quite striking given the simplicity of this molecule. And this, of course, contains just carbon and hydrogen. But the chemistry of life, of course, requires a lot more types of atoms. This is glucose that includes oxygen, a main source of carbon and energy for our own metabolism and many microbial cells. And you can add to it nitrogen, which of course is the essential atom of all amino acids, such as tryptophan. And I'm pointing this out because sometimes the chemistry is so complex that one gets easily lost, like myself, but it's always useful to remember that just by looking even at what atoms are present in different molecules, there is a lot you can figure out what are the demands and then needs of the cells in order to produce a certain category of molecules. So nitrogen is essential for the production of amino acids and proteins. And there are molecules such as ATP that contain phosphorus. This is the 3 phosphate groups that can be hydrolyzed to release energy. And in fact, ATP, as you probably know, is a fundamental molecule that stores energy. So this is the energy currency of the cell. And it's very important because that's how the cell transfers energy between processes and allows the driving of reactions that would be otherwise thermodynamically infeasible. So the other atom that I want to point out is sulfur. It hasn't appeared yet, and it appears in this molecule, which is called coenzyme A. And you'll see, by the way, similarities. This molecule is similar to ATP, and there's this chain that contains now a sulfur molecules. And here we really pretty much completed the main elements that are essential for living systems. There's, of course, more. There are metals and so on, but by and large, these are some of the atoms one worries about when thinking about basic metabolism. And coenzyme A is a cofactor that is used for transferring groups between reactions and is, again, a very widespread molecules across living systems. And I'll jump to a different scale to show you a different type of molecule. This is a protein, a very large multi-protein enzyme complex called ATP synthase. This is a stunning machine. I'm always amazed when I see this. This is a molecule that is made of many, many atoms, as you can see, and it's one of the enzymes that catalyzes, enables the reactions that transform the small molecules we saw before. In this case, this molecule is what enables the production of ATP across the membrane through a process called respiration, which we'll see in a very brief overview very soon. This is in the mammalian cells, but what is amazing is that every single cell that contains these molecules has to produce these proteins in enough amount to carry the reactions, and the reactions themselves are needed to fuel the production of these molecules in a very complex set of feedback loops. Notice, by the way, that proteins don't contain phosphorus. So again, interesting to look at the elemental composition of different classes of molecules. Protein of the mass of our own cells, and they don't contain phosphorus. So one can start asking questions that we'll see later on of when and how different elements took part at different stages in the history of life to make it possible for living systems to jumpstart these metabolic processes. So I want to now jump from this to starting to tell you how and why these metabolic processes really can affect ecology. We want to get to ecology fairly quickly, although today we'll mostly talk about metabolism at the single organism level, but I want to start giving some glimpse of why and how this is so important ecologically. So these are two basic metabolic pathways, and I'm drawing this again in this very simplified way that is the carbon backbone. So these are the number of carbon molecules involved in this first molecule, which is actually glucose. It's broken down ultimately into pyruvate, which is a 3-carbon molecule. And in this case, there is another cleavage, leaving a carbon 2 molecule, which we'll see later is a fermentation byproduct. And in this process, the cells can produce 2 ATPs. So there is the energy currency production is 2 ATPs per glucose that is being broken down. And this is a fermentation process, what gives rise, for example, to ethanol in yeast and so on. Now, there is a different pathway, and many cells can carry both processes. In fact, if you continue feeding some of these carbon 2 molecules in a number of different ways through the citric acid cycle, which is the cycle we saw before, this kind of semi-autocatalytic cycle that we saw before, this process in a much more complicated way, which I'm not going to go into now, can lead to the production of 32 ATPs per glucose that is consumed. So this is an addition that makes a big difference in terms of the production of ATP. And again, many cells have the option of just stopping metabolism here, doing fermentation, or keep going and respire the molecules. Now, of course, things that are much more complex in real life, but this is just to give you an idea for those that are not familiar with metabolism, the kind of questions one can ask and what are the implications. So what are the implications for ecology and how one think about this? So one thing that may be already obvious from this very different yields is that in fact there may be a rate yield trade-off that could be very important for competition and cooperation in across different bacteria. Of course, this kind of metabolism is much more efficient. You produce so much more energy currency per glucose consumed, but there is evidence that this is somehow more cumbersome, potentially slower in terms of rate, and it certainly requires a lot more proteins in order to carry these processes. In fact, this molecule, the ATP synthetase I showed you above, is exactly what allows the production of these 32 ATPs. And what is interesting is also, of course, this requires oxygen, as you might imagine. Respiratory pathways, this is the final electron acceptor for this process. But what is important is that without oxygen or other electron acceptors, this metabolism cannot occur. Cells can run this metabolism in absence of oxygen. And it turns out what is quite interesting and still poorly understood, some cells decide to use, even in presence of oxygen, the fermentatory pathway. And in fact, this is one of the hallmarks of cancer for those of you that are interested in mammalian metabolism. And there is also there questions about the ecology of how different human cells interact with each other and tumor cells. But there is this known phenomenon called the Warburg effect, where human cells, even in presence of oxygen, will decide to ferment. There is a beautiful paper, if anybody is interested in reading more about this, on this rate yield trade-off and possible consequences for the competition between fast but inefficient and slow but efficient organism. And this is this paper by Pfeiffer, Schuster, and Bonhoeffer. There is another important possible impact, ecological impact of the dichotomy between these different metabolic pathways. And this is the fact that, as we said, this molecule that is a carbon 6 molecules could be glucose. The C1 molecule that is secreted is CO2. And typically, the fermentation byproducts are organic acids. These are carbon 2 molecules such as lactate, acetate, ethanol. Lactate is the fermentation byproduct produced by human cells, for example, and by cancer cells. Acetate is produced by E. coli. Ethanol is produced by yeast, as we all know. So what is interesting is that if cells choose to use this fermentative metabolism, they will secrete these byproducts, and these byproducts are perfectly usable carbon sources for other organisms. So you can imagine that the decision of individual species to carry one metabolism versus another can have important consequences in terms of the capacity of interact with other organisms, enabling cross-feeding and exchange of molecules across different organisms. So keep that in mind, and we'll get back to this later on. So the thing I told you a little bit about, right, in this previous slide is how metabolism is important for generating the energy currency, ATP, and also the redox equivalents, which we haven't talked about. But there is another key function that metabolism carries, which is not less important, in fact, very complicated, and it is the production of all the different monomers, the different molecules that are used for producing proteins, the DNA, RNA, and all the components of the cell. So what you see here is a histogram of the proportion of the different biomass components in an E. coli cell. These are the different amino acids, the 20 amino acids, phospholipids, nucleotides, and this is, if you were to take a snapshot of the dry mass of a cell and measure how much there is of each of these compounds, this is what you would get. This is in millimoles per gram of dry mass. So it turns out that the same pathway I just showed you, so you can recognize here the fermentation or glycolysis pathways going from glucose to pyruvate and then feeding into the TCA cycle. So this same pathway, in addition to producing ATP, can be piped along the way to produce other compounds. A lot of the amino acids and precursors for nucleotides and lipids are all coming along these different pathways. So in the same pathway that produces energy needs also to carry the production of all these other molecules. And all of this has to be balanced in a very delicate way because, of course, you need to have the right amount of each of these molecules. It doesn't help if you're very good at producing glycine but cannot produce alanine. All of these are needed in the right proportions. And all of this has to be accomplished while at the same time, the cell also produces the right amount of ATP, because ATP is used for degrading molecules and building molecules. So it's all a very complex balance of different reactions that have to fit together in order to efficiently produce all the biomass components. So you can start seeing the flavor of a resource allocation problem for the cell, and a problem that, in fact, is amenable to mathematical analysis. So we're going to start talking now about genome scale models. But actually, I don't know, I can pause here, just give an opportunity if there is any questions so far. Okay, now I'll keep going. Okay, so we're going to start introducing genome scale constraint-based models of metabolism. And you'll find these models, again, some of you may already know about these. These are known in different ways. For example, they are known as genome scale models sometimes, constraint-based models, or stoichiometric models, and they're all of the above. Usually genome scale because one tries to model the whole metabolic network of a cell, constraint-based because, as we'll see, will rely strongly on constraints, mostly related to mass conservation, stoichiometric because they are based on the stoichiometry of the different reactions in the cell. So one of the first problems in trying to make a genome scale model of metabolism is to construct from this universal metabolism I showed before that contains, again, all the reactions known across all living systems. And now actually the numbers are larger. This is an outdated number. There is probably more than 20,000 molecules that are part of these databases now. But you need to filter this through the genome of an individual organism and ask, for example, which among all these different reactions is E. coli capable of doing. And this is written in the genome. You have to read and see what reactions are present and encoded by enzymes in the genome of this organism, and translate this into a smaller network, which is the metabolic network that is specific for this organism. A typical bacterial organism has of the order of 1,500 reactions and about the same number of metabolites. And this is what is called metabolic network reconstruction. This is in itself now a whole process, partly because we don't know the function of all the genes. So when you try and read the DNA of an organism, there is a lot of unknown genes with unknown functions or partially known functions, uncertain functions. This is a very, very complex set of steps. And it's really interesting. It involves literature curation, sometimes manual curation. There is a lot of efforts now to try and automate this process, but we're gonna not go into the details of this and just assume that we have a genome scale network, the network of all the metabolic reaction occurring in a specific organism, and then we'll ask the next question of how to model this. So if you're interested in where to find these networks, these are some pointers. Bernard Paulson at UCSD has a list of curated models, and he's one of the first to bring this field to really the systems biology field. Model Seed is a database of automatically reconstructed models from genomes. These are all present in the K-base, also an open database from the Department of Energy that has a number of tools for reconstructing models from genomes. And I want to just remind you, if you go into this, that you have to remember that there is a wide variability of completeness and accuracy of these models. Some may have undergone thorough experimental testing for many years and may be very, very well tested and accurate. Others are just straight from the annotated genome, and they may not be as accurate and complete, but it's all still always interesting to have something to start from and to build upon. So what is it that we're really talking about? How do you go now from this network, and how do you represent the network, and how do you translate this into a prediction of what an organism can and cannot do? And I will start from illustrating what used to be, or is still somehow, the classical approach to addressing the question of what an organism can do, which is kinetic modeling. So again, if a typical prokaryotic cell may have about 1,000, 1,500 reactions and metabolites, this involves about 10,000 kinetic parameters. You could write differential equations describing the change of each metabolite in this network as a function of the rates of the reaction that produce and consume that metabolite. And I assume many of you may be familiar with this. This is the Michaelis-Menten equation telling us how the rate of the reaction depends on the amount of substrate present in the incoming molecule, and this Vmax, that is the maximal attainable reaction rate, which depends on the amount of enzyme present for that reaction. And what is important here is that this is a nonlinear function of the substrate. So if you know the concentration of the substrate, you can calculate the flux, but there isn't a one-to-one linear relationship between the two. And at some point, if you keep adding substrate, the reaction will not keep growing in rate, because you might be limited by the enzyme. And the important thing is that you could write an equation like this for every single flux and every single rate in this network, and the real reaction rates would look more like this than like this, because they may have two incoming molecules and two outgoing molecules, multi-substrate, multi-product. So you really have a lot of different parameters and complex nonlinearities that make this kinetic modeling approach very complicated. But we're not going to go into this, and we're going to abandon the kinetic modeling approach for this much simplified version of metabolic modeling, which is going to come next, and is called flux balance analysis. But let me tell you first, briefly, with this example, how do you represent a network? Again, this is an illustration of a simple network where you have metabolite A, for example, in a cell that is being imported through this reaction V1 and produced by a reaction, consumed by a reaction V1, and then also consumed by V2, produced by V3 back from this metabolite C. So you could write for metabolite A this differential equation where there is a term for each reaction that consumes or produces that metabolite. And this is a linear relationship between these different fluxes. But remember that each of these fluxes has this dependence on the substrate, this nonlinear dependence based on the Michaelis-Menten reaction we showed about. So this seems simple, but it's actually a fairly complex differential equation. You can write such a differential equation for each metabolite, and you can easily represent this in the form of a matrix. So you'll have a vector of all the fluxes, a vector of all the metabolites and their derivatives in time, and this matrix really converts the set of fluxes into the changes in metabolites is what is called the stoichiometric matrix of the network, and it's really a compact and very valuable representation of the structure of metabolism. So this stoichiometric matrix is essentially what you need to have as an outcome of the metabolic network reconstruction in order to start modeling metabolism using flux balance analysis. So as I said, this started as a resource allocation problem. It started in the field of chemical engineering from Terry Papoutsakis and others, and brought now to the forefront of systems biology by Bernard Paulson and colleagues. And now it's a very widespread approach, and it's often called flux balance analysis, as we'll see in a second why. This is again a representation of metabolism for E. coli. There are nutrients, and remember, nutrients have to contain at least one source of carbon, nitrogen, phosphorus, sulfur, and so on, all the elements that are needed to produce the different molecules. And then these molecules flow into the proteins and the DNA and RNA and lipids through the construction of the precursor, the monomers that are essential for building those molecules. And the cell, by putting these molecules in the right proportions, as we saw before, produces what we call biomass, new cells that have, again, the right proportion of biomass components. There could be production of byproducts. And what we need to do now is try to find a way to simplify this process. So if I zoom in one of these reactions, this is glucose 6-phosphate, again, the beginning of that fermentation or glycolysis pathway I showed before, with glucose coming in. And there is a very simple mass conservation, which we impose by assuming that the system is at steady state. And this is the flux balance approximation, which is very helpful. Of course, one could argue whether or not this is reasonable. We can talk about this more later if anybody's interested. But for now, imagine that you're keeping a population of cells in a bioreactor in steady conditions, and it seems reasonable to assume that overall, on average, between all the different cells, the net amount of each metabolite overall in the population stays constant. There is no net accumulation or depletion of compounds. And this translates in the fact that now this sum of all the fluxes producing and consuming this molecule is now balanced to provide 0. So this is the flux balance part. And now, remember again, each of these fluxes would depend on the concentrations, but really this is where we abandon the metabolite concentrations and really focus just on the fluxes. And if we just focus on the fluxes, if all we're interested in is knowing what are the rates of these reactions, then we're really just dealing with this linear equation. And this is the world of flux balance analysis, is a world of fluxes where we forget about the metabolite, the internal metabolite concentrations, and we just try to understand the fluxes of these reactions. And of course, this makes this problem much simpler. There is a constraint like this for each metabolite in the cell, but there is also more constraints that you can add. In particular, there are constraints about the capacity of what is coming into the cell. And these are very important constraints because they define the specific conditions under which you're running a certain experiment. So for example, if a cell is grown, if a population of cells is growing in a bioreactor with glucose available, and you know how much glucose you're putting into the bioreactor, the cells could not possibly take more glucose than you're providing. So this will give rise to an inequality in the flux of the glucose intake. Okay? So again, this is a linear relationship, and you'll see in a second how all this, the linearity of the constraints for the fluxes will make it possible for us to have a mathematically tractable problem. There are other constraints. For example, some reactions, all reactions, all metabolic reactions are supposedly reversible, but effectively, some reactions may be so unbalanced thermodynamically that are effectively irreversible at physiological concentrations. And if this is known, one can impose additional constraints on some reactions going only in one direction. So for example, this flux for this irreversible reaction would be said to be positive. And this is, again, another constraint and another linear constraint on the fluxes. So all of these constraints together, the linear constraints for the conservation of mass at each node, at each metabolite, for each molecule coming inside the network, possible irreversibility constraint, they define a space in the multidimensional space of fluxes, which is called the feasible space. And as you can already see, this is a convex polyhedron. Why is this a convex polyhedron if this is not obvious? Well, because you have just hyperplanes, right? Constraints like this are hyperplanes of dimension n-1 in the n-dimensional space of fluxes. And of course, it's difficult to represent this, so I am representing here a projection of this space on 2 dimensions, 2 arbitrary fluxes. So you have hyperplanes, and you have one hyperplane for each constraint, for each metabolite that is conserved. This hyperplane will intersect each other and form subspace whose dimensionality depends on the whether or not these constraints are linearly dependent or not. And when you add this capacity constraints, you take half spaces and you end up having these polyhedral structures, convex polyhedra, that represent the feasible spaces for the cell. Okay, so this is again a simple ideal projection in 2 dimensions of this space. So this is in itself interesting, and there is a lot of work now on just sampling this space. So if you know nothing more, but you have these constraints for intracellular metabolism, you know what the capacity constraints are related to the specific conditions under which you're running an experiment, you can already have this characterization of what the cell can and cannot do. And this is already quite intriguing and quite interesting. And again, if you think about this as in contrast to kinetic models where you have differential equations, you solve the differential equations, you have a specific solution. This has a very different flavor. Here we don't have a specific solution, but we have this algebraic representation of a space of where the cells can be found. So it's an interesting geometrical object that enables a lot of subsequent analysis. And one thing that has become kind of the standard approach in stoichiometric modeling is the idea of using optimization. You might imagine why optimization might be helpful, but if you think of a cell as a system that has undergone long evolutionary selection towards, for being efficient at producing its own biomass, growing efficiently, growing fast, you can imagine that objective functions such as the maximization of the growth rate might be a reasonable hypothesis for what a cell might be trying to do. And the advantage of having an objective function is, of course, that now you can look within this space of possible fluxes, you can look for a flux that is optimal for a given objective function. And for example, if you were to try and find within this feasible space the point that maximizes Vj, this would be the point up here. If you have in general a general objective function represented by hyperplane that you can imagine sliding along the space, and when this encountered an extreme of the space, this will be the optimum for the function. And you can, in this way, find the maximum for this growth rate, which will tell you among all the feasible points for the cell that balance all this consumption and production of molecules, you can find the point that allows the cell to grow in an optimally efficient way. Okay? And this point, what is important about this specific point is that this is now a prediction that one can test experimentally. You'll have as an outcome a vector of all the fluxes of the cell for every single reaction, as well as a prediction of this growth rate. So you'll have a value for each of the fluxes, as well as the value for the growth rate, which will tell you how fast, given all of these constraints, how fast do you expect a cell to be able to grow. And by the way, something that, again, we'll see more next time, but you can also predict whether a cell will produce byproducts. And those byproducts, if you were to somehow embed this organism into an ecosystem with other organisms, that byproduct could now be the source of a cross-feeding interaction between multiple species, which is why one at some point realized that this kind of models can be really helpful for modeling the ecology of microbes. I want to point to some practical resources. If you have never seen this, of course, I have really enough information to get started right away, but I want to point to a couple of things that might be helpful. This is a paper from Jason Papin's lab that I think is a really nice overview and practical way of getting started with doing these flux balance models. I think it has some Python scripts that you can start using right away for doing first simple models for simple networks and then going into increasingly complex networks. There is a very nice Python toolbox called CobraPy. Cobra stands for Constraints-Based Reconstruction and Analysis. So this is one of the names by which you'll find these flux balance models. And CobraPy is a freely available resource for doing all sorts of things with flux balance modeling, uploading models, running optimizations, and so on, and it's quite convenient. This would be a good starting point. I also put here in a GitHub on our lab webpage some basic scripts that I've been using for doing simple FBA and some models, including the human cell model that is now available. And there is it's a big world, so there are a lot of possibilities out there. There is a MATLAB toolbox. There are different resources. Feel free to ask me later on if you need pointers to specific resources, but this should be a good starting point. Now, okay, in the next few minutes, I want to point out some of the applications of flux balance modeling before going back to the ecological side. And you'll see actually that there is an interesting connection between looking at what happens inside individual cells and scaling this up to the ecosystem level. So one of the typical applications of flux balance modeling in the past has been to try and understand what happens if you delete a gene from a network. So imagine having E. coli. You can predict the growth rate using flux balance modeling and ask what happens if you remove one gene, one reaction, say, from the organism. Will the organism be able to survive? And one thing I realized now I forgot to mention is that one of the reasons this method is so valuable and efficient, some of you may be already aware of this, is that, let me go back here for a second, solving this problem is really in itself a very efficient process. This can be done through a number of linear programming packages and algorithms, starting from the simplex algorithm to now more advanced models, that algorithms that use heuristics. But essentially, in a fraction of a second, I think 100th of a second or so, you can have a solution to a single flux balance model. So imagine now, yes, there are caveats and there are things to be careful about, the assumptions we made, the simplifying assumptions we made. But on the other hand, in a fraction of a second, you get a prediction of all the fluxes in the cell. And again, this is why this is, because it's so fast, one can use it to address questions such as doing all possible perturbations of the environment or the internal circuits of the cell to see how the cell responds. And we're going to go back here to this slide, where the process of finding how a cell responds to a perturbation can be viewed as a problem of reducing the space and finding again a point that is physiologically relevant on the reduced space. Let me just give you an intuition for why that is the case. When you have, so this green region represents again the feasible space for the wild type unperturbed organism, where you can find its own objective function. If you remove, and let's leave aside the fact that there may be a complex mapping between genes and reactions, but let's assume for now you just remove a reaction from the network. A reaction is made impossible because of the lack of a mutation into a given gene. So that flux will suddenly only have the option of zero flux. There is no flux through that reaction anymore. So you have an additional constraint in this multidimensional space. So we'll reduce this space to a subspace represented here in yellow. And now you can find within this subspace what is, for example, again, the optimal, the maximal capacity for the cell to grow. And in this way, you can find, first of all, whether or not the cell can still grow after doing that perturbation and how fast. And you can compare and predict all the different knockouts in the genome of an organism relative to each other and relative to the wild type. And again, this enables a lot of different downstream applications, from metabolic engineering to evolutionary algorithms, start to think about evolution of metabolic pathways and so on. There is one thing that some of you may be asking yourselves, and which is a question we were interested in many years ago, which is whether really metabolism should be optimal also for knockouts. And you can think of, first of all, this question of whether or not we know what the objective function is in itself an interesting open question, but it's particularly tricky when you think of a knockout organism. If you remove a gene from an organism that maybe did undergo long-term evolutionary optimization for being efficient at growing, there is no reason for a perturbed organism to be efficient in its own subspace, right? So it's entirely possible that this perturbed organism will not be able to perform in its optimal way, given that you just performed this perturbation. So what is interesting is that you can look at alternative points in this space that may better represent what you expect a perturbed organism to do. And one possibility is to look at the projection of this wild type point optimum on the original space onto the space of the knockout. Why might this a better prediction for the knockout? If you think about this, what the implication of assuming that the wild type organism is tending towards this optimum is that its internal regulatory pathways really allowed it to upregulate and downregulate the different genes to achieve this optimal production of the biomass components in a balanced way. But once you remove a gene, the organisms still have that same regulatory circuit. So it's still tried to go towards this optimum. And you can ask, what is the point that is as close as possible to this wild type optimum, but still constrained onto the space of the knockout? And this would be the point that is at minimal distance on the yellow region of the knockout space that is as close as possible to the wild type within this yellow space. And you can solve this by minimizing this distance. There is no obvious reason whether one should choose a Euclidean distance or an L1 norm or other distances. All of this have been tried. You can use quadratic programming for minimizing the Euclidean distance, and you can find this prediction of the knockout, which turns out to be, in many cases, a little bit more accurate than the prediction of the what the wild type or what the knockout optimum would do. And of course, one could imagine that throughout evolutionary processes, a mutated organism could go from this suboptimal initial point to the optimal point in evolutionary steps. Let's see, I think I have a few more minutes. So this method is called minimization metabolic adjustment and is one of many methods now that people use to probe metabolic networks for under different scenarios. One thing that I haven't told you, and I want to give you a glimpse of whether and how one can test these models, and also the the caveats that one has to keep in mind in making these models. And this is an example from a comparison I did many years ago based on data from Uwe Sauer's lab at ETH. So this is E. coli grown in a chemostat in a bioreactor kept at constant flow. And you see, by the way, you can recognize here again the pathway we saw before, glycolysis and the TCA cycle. And what was done here was to compare experimentally measured fluxes with fluxes predicted with FPA. And first of all, I want to mention that measuring fluxes experimentally is a very tricky and laborious process. Typically, this is done with carbon-13 labeled metabolites that go through the network, and the carbon-13 atoms are dispersed through the network in different ways, and one can then figure out the actual fluxes by mapping where the carbon-13 went. But this is very complicated, and by the way, we know to do this, and not we, experts in metabolic measuring can do this for individual organisms, but it's still an open challenge to try and measure fluxes for communities, and a very important one, as we'll see later on. So, but what I want to highlight here is that there is a good agreement overall, if you compare experimentally measured fluxes with predicted fluxes for this E. coli grow under carbon-limited conditions. So this is one mode of running this chemostat, carbon-limited, and there is very high agreement, which was for me when I first saw this, and now there's a lot of testing of different models. Some work better, some not as well. But this allows me to illustrate the fact that even the same organism under different conditions can have very different degrees of agreement with the flux balance prediction. So for example, if you take the same organism and just compare experimental fluxes with predicted fluxes under nitrogen limited conditions, you can see there's still some correlation, but there's clearly something that we don't understand here, or there is something that the model doesn't predict correctly. And one could speculate on why and what could be going wrong here. Why is the model working under one condition and not the other? And one possibility, for example, is that the assumption of maximal growth rate is reasonable for carbon-limited E. coli, but not for nitrogen-limited E. coli. Perhaps evolutionary adaptation and the regulatory circuits in E. coli are really compatible with his idea and hypothesis of maximal growth rate when there is abundant carbon. When carbon is a limiting resource, but not when nitrogen is limiting, and there may be other strategies that the cell may choose to pursue. So this is one example, and just to illustrate again that I view flux balance modeling as a hypothesis testing tool, as a way of asking interesting biological questions. Sometimes it can be used for valuable predictive modeling for metabolic engineering applications, but one has always to keep in mind that some of these assumptions we made may not be true under all conditions. I think I have a few more minutes. I will point just to other papers that I think are representative of how people tested these models. There is very nice work on showing how adaptive evolution can lead from organisms that are initially suboptimal under a certain condition to gradual optimization. There are other work showing that this is, again, not always necessarily the case. This was from Bernard Paulson's lab. This is from Chris Marks and Will Harcombe. This was actually based on data from the Lenski evolved E. coli lines. Very, very interesting work. So this is, there is now a lot of work using these models, and you'll see more, and I bet you'll see exciting work from Alvaro Sanchez's group that also has more of an evolutionary flavor. I will last conclude just by mentioning some of the good and bad aspects of flux balance. So I think I wanna highlight again that there are some really valuable and good reasons for using this modeling approach for a number of applications, including, as we'll see, looking at ecological interactions. It's very fast, it's very scalable. You can easily look at larger organisms, multiple organisms together. And very importantly, you do not need the kinetic parameters, right? Once we made this transition from the world of metabolic concentrations, metabolic concentration to metabolic fluxes, we really forgot about the metabolite concentrations, and therefore the steady states we compute we don't really need to know the kinetic parameters. It gets more complicated, as we'll see later when you want to go to communities, but we'll leave this for next time. But one thing I want to highlight, which again is on the positive side here, is that concentrations are obviously important, but there is something really unique about fluxes, and I think the cells care about the fluxes. We care about the fluxes. If you want to know how much is produced of a given compound, how much is consumed, this is going to be super important for ecological interactions. So it's somehow fortunate that these models, through these simplifications, are good at predicting fluxes, because they will be very helpful for embedding the single organism model into ecological models. But there are some limitations, and sometimes you really would like to know metabolite concentrations inside the cell, because that's what is more easily measurable now with metabolomics approaches. And because of the lack of metabolite concentrations intracellularly, you cannot really explicitly model regulation, because regulation, in particular allosteric regulation, where small molecules bind to the enzymes, for example, this is strongly dependent on the internal metabolite concentrations, and this is really beyond what flux balance models can do. There is very interesting work now being explored, where if you incorporate some thermodynamic constraints in these networks, it's possible to put back concentration of metabolites. But I think that's where the field needs a lot of creative energy for people trying to think how to go beyond the kinetic paradigm and the flux build maybe hybrid models that have the best parts of both. There are other limitations. You cannot easily model fast dynamics because of this inherent dynamic steady state approach. And what you predict is really population and time averages, not single cell fluxes. The other thing that is important to remember is that how accurate these models are will depend also on how well you know the boundary conditions, for example, the uptake rates of different nutrients. Some of the challenges, some of the open directions, which again will be relevant for ecology as well. I showed you this snapshot of the biomass composition of a cell, and this is often treated as a fixed vector of numbers, but in real life, this is a condition-dependent composition, right? E. coli and all living cells will change their biomass composition as a function of the environment. There are striking examples of marine bacteria that instead of phospholipid, use sulfolipids when they're under phosphorus limited conditions. So this is really important, and we barely know, how the biomass composition of cells change. So this is very interesting. We know very little about the maintenance, how much energy is spent by metabolism in doing non-metabolic processes. Oops, sorry. There is interest again in, as I was saying, in mapping the global effects of thermodynamics of these networks. But this is an ongoing challenge, partially because we don't know the chemical potential of a lot of these molecules. And the last and not least important challenge is that, as I said, we know quite well the models for some organisms, even despite the limitations. But when you start thinking about extending these approaches to modeling a whole gut microbiome with thousands of different species, or the ecology of microbes in soil, this becomes a much bigger challenge. And what a lot of people struggle with is how to efficiently build models for all these different species and scale up these modeling approaches to really start looking at interaction between species in complex communities. And this is where we'll start from next time. So I will pause here, and I think we have time for questions. So thank you.
Thank you very much, Daniel. Yeah, I think we have a raised hand from Ashish George. You can ask the question.
Hi, hey Daniel.
Yeah,
that was a very interesting talk. I had a question actually about the first part in terms of like this kind of acetate crossfeeding in E. coli, or like the respiration fermentation kind of thing. What's so special about, I guess, acetate, or I guess stopping in this point just before the TCA cycle? Like, why not crossfeed somewhere else? Why not in between the TCA cycle or a little further above? Is there something special about this point that E. coli likes, or like cells like to?
So that's an excellent question, and I think this is still a highly debated and hot area of research. I'll give you an idea. For example, one of the there is one of the classical examples, which is true both for yeast metabolism and for cancer metabolism, that we really don't know why certain cells, despite having the possibility of doing the TCA cycle and despite having oxygen around, choose to ferment. So for yeast, for example, one of the hypotheses is that in a competition for survival with other organisms, it may actually be beneficial to stop here, secrete ethanol. As you know, ethanol is a good disinfectant, right? So it can kill bacteria around. So it might be a good strategy for yeast to first secrete the ethanol, kill competing organisms. And in fact, yeast is capable of taking up the ethanol again, doing the respiration of ethanol after consuming all the glucose. So this is called a dioxic shift. And so there is a lot of complexity in this processes where cells may decide to do first the first half of the pathway and then retake up the compound that was secreted and use it through the complete.
Okay, so
respiration.
So like acetate and glycerol or something would also could have inhibitory effects. Like, I guess like E. coli shows like excretes acetate or glycerol or something, right?
Yeah, so let me tell you something else that happens. So again, this can vary very much from system to system. And you know, people have come up with different reasons for why organisms sometimes ferment, even if they could respire. But in this, let's say in the case of acetate for E. coli, for example, what might happen is that E. coli might be actually oxygen limited. And if you're oxygen limited, right, you cannot run this pathway, or you can run it only partially. So it is possible in principle that when you're limited by oxygen, you know, you have no option but to run this pathway to keep going, and then you produce acetate, and if oxygen becomes available, you could in principle respire. But in other cases, such as the lactate production for cancer cells, is really highly debated, and there are many different reasons for, you know, people believe this might be happening, one of which has to do with efficiency, and again, this trade-off between rate and yield. I think this paper I was mentioning by Pfeiffer, Schuster, and Borhofer has some really interesting hypotheses about the fact that really maybe there are some deep thermodynamic reasons, and there are some old papers proposing this, that fermentation might be indeed inherently faster than respiration. So really there is a trade-off between rate and yield, and you can imagine that if there is a population, you know, the fast but inefficient will take over typically, and that would cancer cells might do, whereas in the slow but inefficient, the slow but efficient organisms or cells would be able to survive in the competition with the fast and inefficient if there is spatial structure. So this is one of the hypotheses of this paper, and it's potentially a hypothesis that could explain, for example, the competition between planktonic cells, cancer cells, versus the structure of the body that is based on this efficient, well-organized metabolism, but also about the rise of multicellular organisms. And this all seems to match in the sense that, you know, when oxygen becomes available, you can do this more efficient metabolism. There is a more, cells are more thoughtful about using resources efficiently, enabling the rise of complex multicellular systems. So I don't know if this addressed completely your question, but the complete landscape can be very complicated, and some of this has to do with, you know, deeper biochemistry reasons, which we cannot go into now, and, you know, I'd be happy to talk about.
But thank you. That was good.
Okay, we have a question from Martina Darbello.
Hi. Thank you for your talk. And so could you come back to the slide that we were talking about the usable byproducts?
Oh, this one?
Yeah, so my question is, so here it seems that cells, let's say, release in the environment usable byproducts that can be used by other cells only if they ferment. But isn't it a bit of a huge assumption?
Thank you. Yes. So this is definitely not the case. So this is one way in which cells could secrete something that is usable by other cells, but it's by no means the only option. I think this is observed often, but there may be many, many, many different ways in which cells secrete byproducts that are used by other organisms. And we'll see this extensively next time. But there is a lot of, if you measure the molecules that are spilled out of a cell, they can be very different and complex. There is also a whole really interesting and, again, fairly open question of how much of this cross-feeding happens through byproducts that are secreted by live cells, as opposed to cells dying and spilling out everything they have inside. So in that case, everything that a cell has inside becomes usable byproduct for other cells, with huge ecological implications, definitely in the ocean, cycling, and so on. So again, I think you point to a very interesting questions. You know, in some cases we know, but in most cases we really are just starting to scratch the surface of mapping this usable byproducts.
Okay, thank you.
We have one more question from Monday, Sunday, Adirha. Please.
Yeah, please. So you talked about limited oxygen in the two pathway of the fermentation and the respiration, but I'm looking at something. If there's a limit in oxygen, would this pathway still exist, or there can be a reverse or a distortion?
Okay, great question. So the pathway itself, right? If an organism has this pathway is there, meaning that the proteins for making this reaction happen, they are in the genome, they're staying there. And of course, the organism, in absence of oxygen, could decide not to express those proteins that may not be expressed. Although, as you remember from this slide, right, there are other reasons for running the TCA cycle. In fact, the cycle itself can run even in absence of oxygen. What needs oxygen is some of the downstream processes. But in order to produce these byproducts, the cell may need to run some of all of the reaction in this cycle. But it's also true, and I think this is what you're hinting to, that these pathways can also run in reverse. And the diversity of metabolic pathways across microbes is incredible. And some organisms will have some portions of this cycle. Some organisms, and for example, if you feed an organism ethanol or acetate, they will not have the glucose that is necessary for building other molecules, so they will run this pathway backwards. So I think different organisms will have different options, and they could, depending again on whether, that there is the two aspects of this. One is what capabilities they have in their genome, and some may or may not have all of these capabilities, but even if they have the capabilities, based on the presence and absence of oxygen and through sensing and signaling and so on, the organism may decide and figure out which enzyme to express. Does this answer your question?
Yeah, thank you. Thank you very much. I'm okay.
I don't see any more questions, so maybe it's time to take a short break before we start with our new lecture. Thanks again, Daniel, and see you soon.
Thank you. Bye.
And we'll make a 5 minutes break, more or less, and reconvene again at 6:15 CET time.
May I open the breakout room? Yeah.
Antonio?
Yes.
I just wanted to check. You can hear me. The audio.
Yeah,
okay?
I can hear you well.
Okay, thanks.
Sure.
And I can share.
Yeah, if you want to check, share the screen, sharing.
Yeah.
Yeah.
The one difficulty is that you can't. My face that you're seeing is not the one that's talking, unless people pin the video. But to do with I'm doing it from an iPad and so on. So
if I'm muted, it should just appear. So if I'm muted.
No, but it'll show me on my with just a picture, not me talking, because I'm on there twice, and the other one is the one that you can see. But because of some feedback issues, I can't have that one being the one that picks up the microphone. So anyway, it doesn't matter. I'm not sure if I can see the chat. If things in the chat right now, it's empty. Shit.
Can you see the message in the chat now, Daniel?
Yeah, but I somehow got moved to a breakout room when I tried to send.
Oh, okay, okay. You should.
Oh, leave room. Okay, I can do leave
leave the
room.
breakout room and stay in the main.
Return to main session. Okay. Okay. Yes, I'll try to keep track of if there's questions that come up in the chat that can be addressed right away. So.
Sure. Okay, welcome back, everybody. We are live again, and it's my great pleasure to introduce to you Professor Daniel Fisher from Stanford, who will give 3 lectures on evolution and ecology in high dimensions, what should not be surprising. Thank you very much, Dan.
Okay, thank you. So I hope you can hear me. Yes. So I want to just start with some caveats. I'm a theoretical physicist, not an ecologist. I've worked only a tiny bit in things that are relevant for this school. So I would ask people if there's things that I say that are blatantly wrong facts to correct me. However, I reserve the right to dispute what's a fact. If you could please ask questions in the chat, and I'll try to keep an eye on it. So if there's questions that are of immediate clarification, then I'll try to answer them right away, and the other ones can leave till later. If something seems to need clarifying and you want to unmute and ask quickly, you can also do that. The second just apology is because of setup and feedback issues. There's two pictures of me. One of them is the one which has a picture of me actually talking. The other one, which is the one you'll see if you're on the speaker, which is of me, just a snapshot. If you'd rather see my mouth moving, you can put that one on, and you can pin that one. Okay. So I'm going to do a mixture of sort of pre-prepared things and not pre-prepared, and so I hope that will go more slowly than if I just write things out. So this is very much a theoretical talk, except for some things I'll show at the beginning for motivation, and I'll explain what I mean by the subtitle of what should not be surprising. So the general focus is going to be thinking of biology in the real sense of being high dimensions and the role of randomness in modeling. Can anyone see my screen now?
I think your audio cut out for the last 15
Yeah,
seconds.
can you hear me now? 5 seconds.
Sorry, you can hear me now. That's okay?
Yes, no, it's okay.
Okay, for some reason my computer one seems to have cut out. Okay, there it is. Sorry. Okay, sorry. So that I'm going to add ecology, and then the second and third lecture, I'm going to come to something which has already been introduced by many speakers, particularly by Stefano Alcina, of talking about the ecology in high dimensions in the sense of Lotka-Volterra models with large numbers of species. And then I'm going to add a crucial part to that, which mostly raising questions about the evolution on that. Okay. So first, some of the questions and motivation. Okay. So one of the key questions: why is there so much biological diversity? That's obviously a big general theme of this school. And the classic example of beetles. There are various factors that contribute a lot to being able to have that. There are many different niches. There are many different environments. And they geographically separated. What does that mean? That means they're not all competing with each other. Okay. Well, one of the things that's become clear in recent years, and becoming more and more clear the more people dig into it, is that the diversity really goes down to much smaller scales. And I'm going to show one of the most dramatic examples. So this is bacterial diversity within a species. And this is a species of Prochlorococcus. It's a cyanobacterium that dominates the photosynthesis in the tropical oceans. And this is showing a tree of from 96 single cells in a very closely related subclade of this species. And there's a tremendous amount of diversity here, and more data with more and more cells, so that even from the same sample, and these are all taken from a few samples, even from the same sample, you see basically every cell is different genetically. And the time since the most common ancestor of this whole group is about 1 million years. Of the smaller subclades of this is about tens of thousand years. And these are all mixed together. They're free floating in the ocean. And in fact, they're mixed together over the whole ocean in about 100 years, or globally in about 1,000 years. So this is extensive work of Penny Chisholm's group in this particular paper by Kashtan and collaborators. Okay. So that's really dramatic, and that's something which we'd like to try to understand. Surely there are not so many niches here that we can explain every possibility with a niche. So we'd like to ask about general questions about the diversity. Okay, so a question we'll want to ask is whether or not sufficient organismic and environmental complexity in a general sense implies that one should expect this very kind of extensive diversity, with just enormous amounts of diversity all the way down to within species, subspecies, and so on. And particularly in the most dramatic case where everything is mixing together and competing. Okay, so that part I'm really going to talk about in the next two lectures. Today I'm going to mostly start off with the other part, which is about evolution in simple environments. And so this is really intentional experiments done in the lab, where they're trying to make a very simple environment where there's no ecology. Everything is well mixed together. It's E. coli in low glucose, so the selective pressure from the low glucose. And this is a very long and beautiful experiment carried out by Rich Lansky over, I guess it's now more than 30 years. And what they find is that the E. coli evolved. There are many different genetic routes to higher fitness. Even in this very simple environment, the E. coli find many ways to do better. Now, when this experiment was started, there was essentially no DNA sequencing at all. And the kind of things that have become possible with this experiment in recent years, from the enormous advantages of the sequencing, have meant that one can go back and look at the fossil record and try to look at the fossil record and track the evolution that's gone on. Okay, so the particular thing I'm showing here is the dynamics of the frequencies of the mutations. So this is the fractions of the population that each mutation has, and the mutations as they arise. And generally, over here at this end, the mutations, a lot of mutations arise quickly and take over the population. So this is one line, this is one flask, there's always shaken, so there's no geographical structure. He's done many different flasks in parallel, and they all go somewhat differently, and some aspects quite differently. So these mutations come in quite fast. And then, not surprisingly, it's sitting in a fixed environment. Gradually, evolution starts slowing down. The evolutions come up more slowly. They tend to come up in groups. We have theoretical understanding of that. Okay. But then, after 10 years or so, something sort of funny happens. You get things coming up and coming back down again, and then some of those sort of swerve up and down over here. And there's a whole sense in here as to what's going on. And it's clear that there's something in there, which is that they're interacting with each other. There's more than one type at a time. And so this is really indication of the development of ecology, even in this very simple conditions. And that's seen that the ecology that develops is different in the different runs of the experiment. Okay, but then, and this is the bit that I want to just focus on now, then something happens later on. It seems to go back to being relatively simple here with sweeps coming up. Okay, but the weird thing that notices is that the sort of rate at which mutations come up and take over here is not that much different than what it was much earlier on. It's slower than it was at the beginning, but it's not different much later on. If you try to compete these with the original ancestor, they may be doing slightly better, but not much. And it's not clear if that can account for the rate at which they come up and how fast they come up, how come up they there. So this is a real question mark as to what's going on here. I'm not going to claim to really understand it, but I want to use that as a motivation. Okay? Because this raises the possibility of whether even in a constant environment, so in a constant environment here, whether or not the evolution can continue forever. Okay, so the question we want to ask here is whether evolution in a constant environment can continue forever without slowing. And I really want to put a can in here. I put a can at the beginning. Can evolution in a constant environment continue forever without slowing? Okay. So this is a thing which we're going to address from theoretical considerations today, and make some argument about whether or not we should expect this or not expect this. And therefore, is it something which is intrinsically surprising or something which is not so surprising? So I want to, but before going into that, I really want to sort of think about the sense in which I mean by high-dimensional biology. So if I think of within the cell, then we've got what we could call the nanophenotype. So the nanophenotype, which is the properties of all the proteins and how the proteins bind to DNA and so on. Okay, and that's some very high dimension. A very high dimension, at least tens of thousands, even for a simple cell, and already substantial for a bacteria. So that's the sort of dimension of the phenotype. That's all the different ways in which the organism can change. Now, of course, that's driven by the changes in the genotype, But the change in the genotype is something which doesn't directly couple to the environment or to the properties. So we're going to want to work entirely in terms of the phenotype in order to try to understand what goes on. And then, of course, one can try to connect that to the genotype, genotype as well. So then this nanophenotype, this codes for the organismic phenotype, which is still something very high dimensional. It's the way, all the ways in which it interacts with the environment, affects the environment, how it's affected by the environment, how it interacts with other organisms, and so on. Okay, so we've got the organismic phenotype still very high dimensions, but the space in which the evolution is working is really of this nanophenotype of the at the microscopic level of the proteins. Okay, and then of course we've got the environmental. And the simplest way to think about this would just be, say, the number of chemicals which affect the organism and the number of chemicals that with it can affect the organism. Now, as Daniel Segre just said in the question at the end of the talk, that if you took each cell and lysed the cell, and so all the chemicals in the cell came into the environment, which others could use or could be affected by, could be poisoned by, and so on, that's something which already is very complicated. That surely is going to give all kinds of roots for evolution, potential evolution, and is something which unfortunately no one seems to have really done that experiment carefully of trying to evolve in those conditions. But the one thing we can be sure of is that the environment is very high dimensional, a very large number of chemicals, and the phenotype is very high dimensional. Okay, so what we want to, what I want to do is try to be motivated by this and think about the consequences of the this high dimensional environment. And let's see how far. Make sure I've got, yeah, okay. So I want to think about the consequences of this high dimensional environment. So what we want to consider is organisms that, already well adapted. They've been around for a long time, so we're not making artificial organisms usually. Okay, and so then we have to think about the fact that everything is conditioned on the evolutionary history. Okay, so the evolutionary history of the organisms, and of course that includes the history of the environment. What is that going to imply? Well, if they're already well adapted, that's going to imply any changes. So changes, which can be genetic changes or changes in the environment. Okay, genetic and environmental changes, their effects will be a sum of positive and negative parts. Okay, well, if you have a large number of positive and negative parts, why do I say that? Well, supposing those genetic changes always had positive contributions, then it would have already fixed in the population, something which is universally positive. So whether it's positive effects or negative effects will depend on the environment. It'll depend on the genetic background, the rest of the evolution that's occurred before. So this is something which we can generally expect as a consequence of the evolutionary history. Okay. So then we can take just a sort of guess. Well, if it's sort of sum of positive and negative parts, it makes it very unpredictable, and this will give rise to something which we can try to approximate in a way, approximating by some kind of randomness, and then look at the consequences of that. Okay, so this already, you've seen someone thinking about the interactions as being complicated. They can sort of be effectively random, but I'm going to really take that and try to run with it somewhat. Okay, so what is the hope? Okay, well, the hope comes from the fact that we have high dimensions. Okay, so this very general quote of Phil Anderson's, "More is different," that when you've got a large number of things interacting together, so something the general character of high dimensions, then things are different than you would expect from looking at small numbers interacting together. There are certain kinds of behaviors. Some of the behavior is independent of details, so we want to know what the behaviors are that can occur. Behaviors in a loose general sense, or the possible things are that can occur. And one of the things that we've learned from physics is that not all the details are going to matter, so we can hope to get things from simple models. So not all the details matter. Okay, in particular, we're interested in behaviors that are sort of robust they don't depend upon all of the details. And that's a sort of loose term, and I'll say a bit what it means in some particular particular context. Okay, so I would like to try to get a robust understanding. Okay, so this really comes to the sort of goal of these talks. So what we're going to try to do is look at some very simple simple models where a key feature is that they're high dimensional and the interactions and things are complicated enough that we can approximate those as being random. And of course, we then have to go beyond that. Okay, so those simple models, we want to then what can exist, what things can occur. Okay, now this doesn't mean that it applies in biology at all, or that it applies in ecology in any particular system, but it's asking what can occur. If we see something that can occur in a simple model, that suggests that seeing it in nature should not be surprising. Okay, now we have to be a little careful there, and that's where this robust part comes in. If we can sort of argue that that can sort of robustly happen in sort of a class of models, a range of models, then we can say some more about, well, okay, that wouldn't be surprising if we saw that. We don't fully understand it, but we get some general sense of it. Okay, now this is in a context which is going to be looser. We can sort of think of phases, meaning phases like a solid or a liquid or a superconductor. So the analogy with phases in physics, and we would like to ask what kind of phases can occur. Okay. And obviously we can't ask about that generally, but we're going to talk about particular. I want to talk about particular examples. Okay, so that's the general scope and sort of what the goal is going to be. And now I'm going to go to some specifics. But before I do that, maybe I can pause if there are any questions on sort of where we can try to go. Okay, Antonio, can you just say something so I'm making sure I at least can hear?
Sure, I can hear. Okay. Well, I don't think there was a question.
So not questions at this point?
No.
Okay.
Oh, okay, there's one question just now.
Oh, yes.
Can you read that?
Yes. This is just to clarify, when you say what can occur, do you mean behaviors or patterns arising out of the nanophenotypes? So yes, I mean things that can come from the interaction between the nanophenotypes as manifested in the organismic phenotypes and the environment. And I'm what can occur, like, for example, do we expect extensive diversity with very large number of closely related strains that can coexist? Okay, and then also the question of things, can evolution continue forever if the environment is constant? So that's the kind of thing that I want to be able to do. Okay, so we're going to now consider an idealized system. We're going to consider evolution in the phenotype space. Okay, so this is going to be of all the properties of the organisms, and I'm going to say this is D-dimensional, which like D was the dimension of the phenotype. I'm going to drop the phenotype for now. And my phenotype then is going to be given by some, characterized by some vector, which is all the D properties of the organisms that will matter. Okay, and then I'm going to make some things incredibly simple. I'm going to make, there's just at any given time, there's just one strain. Okay, no spatial structure or variations in the environment. Everything is mixed together. Everything competes with each other. And then my fitness, in that case, I can make well-defined. It's just going to be some function phi, which is going to be a function of the phenotype and the environment. So this is a vector describing the environment. Okay, it's going to be a function of the phenotype and environment. And this is just going to be the relative growth rate. Okay. Now, I personally, I hate the word fitness. It seems like it's a singular term. It's usually used as the fitness. This is fitter than that. Okay. Well, it's, even though it has two S's, it really is a very parallel quantity. And I've considered trying to get into the literature the idea of a fit nome. I hate the word no, things ending in "ome" generally, but I think this is actually illustrative of the complexities going on. And because at the very least, even when the environment is fixed, of course, everything depends on the environment. If I change the environment, then things are going to change. Okay, so that's the quantitative graph, but we're going to fix the environment, at least for now. Okay, so then we get a small mutation, a small effect mutation, and that mutation will take x to x plus dx, and I'll try to keep doing the vectors at least for a while. I'll get lazy. I'll get lazy shortly, and then if the phi of x plus dx in this environment is bigger than phi of x, okay, then that mutation fixes. So the mutation takes over. Okay. So what does this mean? It means that I can treat my dynamics as being approximately deterministic if the dx is very small, so that the evolution is now approximately deterministic. Okay, and where will it go? Well, it will just go uphill. So it just goes simply uphill in this fitness landscape. Okay, so my dx dt, the way in which it changes with evolution, okay, that's just going to be the gradient with respect to x. So the gradient with respect to x of phi. I'm fixing the environment, and so that's not changing. So I've got a very simple thing, and then of course, if we're doing this for the fixing environment, then this phi is just the landscape that I've got. This is really evolution in a continuous landscape. This is the simplest possible model. Okay, so then of course things are trivial, or at least seem to be trivial. Okay, so I can of course draw the behavior in the case where dimensions are 2. Some people can draw things in 3 dimensions. I can't. And so what I've drawn here is contours of the landscape. So these are contours of phi, and then in those contours I will get dynamics. So if I start at some point, here, I'll come up here. I'm going up the gradient, so I'll bend around like this, and I'll come to a fitness maxima. So I'm going to go up like that. Of course, if I started over here, then I'll come up like this. I'll come up here and go over to this maximum. If I came here, I would come up and go and by mistake erase my contours, which is cheating, and I would go over in this way there. Okay, so here I've got 2 possible maxima, and these are separated by a saddle, which is sort of the decision point of what goes on. And depending on whether the saddle it comes to one side of that or to the other, then it will go to one of these maxima or not. And of course, I can have many maxima, but the thing which we know is that at long times, as we get long times, the evolution will go to one of the maxima. Okay. There was a question when I say relative growth rate, what is it relative to? Is genetic drift important? So the first one, what I mean is that it's growing faster. Its higher fitness will mean it grows faster with something with lower fitness. Okay, so the only things that really matter are the fitness differences. So it's really say the difference between this fitness and this fitness that matters. That means this will grow faster and will take over. Okay, I'm going to ignore genetic drift. One can consider the effects of genetic drift. If we're in bacterial populations, particularly say if they live in the ocean, the populations are very large. Drift is not very important, even though possibility of extinction is important. Okay, so that I'll talk about next time. Okay, so this is the behavior in 2 dimensions, and the general assumption and the way in which most talk about evolution in simple conditions goes is that there are some number of fitness maxima, and long times you go towards the maxima. So the question is that what really happens? Okay, so we're now going to want to consider things going in higher dimensions, but let me first show a bit of the behavior, the complexes can happen in 2 dimensions. So in 2 dimensions, I can look at all of the stationary points, all of the points at which there's no dynamics. So those are the points at which x dx dt is equal to 0. So those stationary points, they can be maxima, as I've already shown. There's a maxima. They can be saddles like this one here, or they can be saddles of index 2. And the index of these points is just the number of the unstable eigenvalues. So a maxima is 0, and a one which is a minimum, this is the minimum in 2 dimensions, is 2, and the intermediate one is 1. Okay. Now, if we go to high dimensions, so we now go to high D or general D. Okay, so then we can get all possible indices of the saddles from I equals 0, which will be a maxima, all the way up to D. And of course, the expectation would be that one would go towards these maxima at long times. Okay, now what am I going to consider? What kind of landscapes I'm going to consider? I'm going to consider landscapes where they have lots of maxima. It's going to be the complexities of the biology, the complexity of the evolutionary history, the evolutionary constraints, and so on. I'm going to assume that phi is a some complex landscape. Okay, and I'm going to approximate this as some random landscape, and I'll be specific about it. Examples shortly. Approximate this as random with some statistics. Okay, so this is meaning that the number of extrema. This is the number of stationary points. Sorry, I was calling that. Number of stationary points goes exponentially with the dimension, and that coefficient then will depend on I. If I look at the number with a given index, the exponential and dimension, and all possible indices will occur. Generally, the maximums will occur at higher phi, and the minimum will occur at lower phi. But of course, I can have local maxima and local minima, which occur at intermediate phase. Okay, so one of the general features which is known from mechanics is you'll get exponentially many maxima and minima and so on. So, okay, one would still expect that the behavior would go to the towards one of the maxima. Okay, so what actually happens? What happens in the limit of high D is if we look as a function of time. So we can look as a function of time. If we look at the function of time, and we can look at the phi. So what happens is initially phi will go up fast, then phi will go along and it'll saturate. So it'll go very slowly upwards. So that's phi. Okay, I can look at the index. Coming here. The index will start out being of order d over 2, and the index will gradually come down. Okay, and what happens here is that the behavior is that it wanders around the saddles. So it wanders around the saddles, all the saddles. It's generally getting to lower and lower index saddle, meaning they're getting closer to maximum, but they always have some unstable directions. And the crucial part here is that it never in the limit of infinite d commits to a maximum. Okay, so there is no sense in the limit of very high dimensions that it approaches the maximum. It always keeps wandering around the saddles. It's as if in 2 dimensions, which of course I can't get this. It came from this saddle, it wandered to that saddle, it wandered to another saddle after that, wandered over here, went to some other saddle, and so on, and just kept wandering around the saddles. And of course, it can't do that in 2 dimensions, but in high dimensions, it can do that. And that's the generic behavior. So this is the generic behavior that happens in high dimensional complex landscapes. This is the generic behavior. It's robust under specific assumptions about what the dynamics, what the landscape looks like. Okay. So that's already a surprise, is that one's intuition about low dimensions is wrong when it comes to high dimensions. Okay, so here the evolution continues forever. It doesn't commit to a saddle, but it still slows down, right? So it's definitely still slowing down here, right? The fitness is going up slowly. The index is going down slowly. If I think in terms of the mutations will fix less and less often. It's harder and harder to find the mutations. So evolution will slow down. Okay. So of course, not surprisingly, the answer to my question of whether evolution can continue forever without slowing down is no, in a constant environment. But now what we have to do is we have to add in the ecology. Okay. So we have to add in a little bit of ecology. Okay. What does that mean? Well, any organism changes its environment. Okay, so if you have phenotype X, so that's the whole population, it's just one strain, right, with phenotype X. What that will do, it'll take the environment to being, an environment will be equal to some function of that phenotype of the organism which is there, and the external conditions, the things that you're holding fixed. Okay? So these you're holding fixed in the experiment, but you can't hold fixed the effects of the organism, because of course if the organism mutates, if X changes, then it will change. Okay, so it's simple then to think in terms of the fitness of the organism when it's the fitness as a function of X. Okay, so that's the function of phenotype. Okay, and then it's going to be the E, which is this E here. So it's now going to be E given by Y. Okay, and then this I'm going to call a different different quantity. I'm just going to call psi of X and Y. Okay, and this X here, this is the resident organism. It's the one that controls the environment. Right, this is the one that controls the environment. And this one can be any. So any other organism that comes in can have this fitness. If this is larger than the resident, right? So if this is larger than the resident, the fitness, then the that can invade and come in. So again, I look at a mutant. Okay, so I start off with X equal to Y. It's just there. But then I look at a mutant, which is X plus dx, and I ask if this is higher. So if psi X plus dx and this fixed Y, okay, is greater than psi at X and Y, and of course this is at Y equal to X. Right, this is at Y equal to X, is the resident that's there, then it invades, then it comes in, and this takes over. Okay, and my environment changes and my fitness changes. So it takes over, the environment now goes to, I get now the environment becomes E of this X plus dx. Okay, so what does that imply? That implies that the dynamics is now going to be driven by these changes, and so the dynamics is dx dt. This is now the evolution. Is dx dt is going to be gradient with respect to X of psi at X and Y, but I evaluate that at Y equal to X, right? Because that's the thing that's already there. Okay, so that's going to be my my dynamics. This is my dynamics here. Put that in red. That's my dynamics. Okay. Now, an important thing about this is not gradient flow. Okay, so this dynamics here is not gradient flow. It doesn't just go uphill. Okay. If you like the physicsy things, you can think of it as some curls to it. It doesn't intrinsically take psi up. What it does is it takes psi up at fixed a value here, right? So it's increasing here. The fitness is going up, but then that'll change it. So it's then going up a different way. Okay. So now what this is going to look like. Let's move this. Okay. Oh damn, that's not working. Okay, so let me look at what happens here. I get flows coming around a saddle. So if I, for example, have my flow here, so now I'm sitting at fixed phi. I now say get a flow which comes like this. So I'm starting getting a flow. It gets near the saddle. It has to decide, right? So it bends over in this this direction because it's that side of the saddle. Okay, but now what can happen, of course, is the environment can change. As it's going along, the environment is changing. So if I do this correctly, my environment is now changing. So my environment changes here. My saddle has moved. Okay, I'm doing it, of course, as a discrete step there. It isn't really a discrete step, it's continuous. So what does that mean? That means that this now goes a different direction. Okay, so the environment changes can qualitatively change where it goes. Okay, so I've got my environment change as it's evolving, driven by the evolution. Okay, gives you qualitative changes. You go in different directions. So this is a picture in 2 dimensions. Again, I can't do things, they don't get very interesting because I've only got small number of maximum I can go to and so on. Okay, so there's a question. Did we assume separate timescales for the ecology evolution? Thank you very much. Okay, I should have certainly said that. We're assuming all the way through this, and I should have said it right, right at the beginning, is that the evolution is very slow. Evo is very slow. Okay, and the environmental changes are fast. With the evolution fast. This means evolution slow in the sense of rare mutations. Only one mutation is coming in at a time. Okay, and the crucial bit here is this idealized, the fact that I said it's always one strain. It's one strain except in transient when I get a new mutation coming in. Yeah, thanks for that question. Okay. So what we want to ask is what will happen in high dimensions. We've now got something which is more complicated than this, because as we're going up here in phi, then the whole function is changing, so I'm going up somewhere somewhere different. Okay, so we want to ask what happens when we've got this. Okay, so what happens if we're in high dimension? In particular, we would like to ask what happens with minimal ecology. Okay, so what is the minimal ecology meaning? I'm going to put small changes in eco, small changes in the environment, Okay, and the specific thing that's going to mean is that if I look at the sort of scale of the gradients with respect to x of psi, so this is the change with the phenotype, okay, this is going to be much, much larger than the typical scale of the gradients with respect to y of the psi. Okay, so this was the environment part, environmental change, and this was the phenotypic change. Okay. And in particular, I can make a ratio between these. So I'm going to say this is much bigger. This is going to be, this is going to be here, say, of order some parameter here, which I'm going to call delta. Okay, so it's going to be of order that, where I'm going to be considering delta much less than 1. So what I want to ask is I want to ask a minimal effects of ecological changes. And I want to ask what happens when delta is small. Okay. Well, in this 2-dimensional example, it's clear what happens when delta is small. If delta is small, then the amount that I'm going to move the saddle by as this comes up is going to be very small. So only if it was extremely close initially. If it was extremely close initially and this changes a bit, will it go in a different direction. Okay, so here it can change it. It'll make little changes where it goes, but not much. Okay, so I think this is, again, the sort of conventional view. Okay, of course, they change the environment, particularly bacteria. They're going to change it by a little bit, but it's not going to be very, not going to be very interesting. Okay, so what will happen if in high dimensions? Okay, so what happens is that the phase that comes in, I'm going to call red queen phase. Okay. And this red queen phase occurs for any arbitrarily small delta. This occurs for any delta, no matter how small, in the limit of d to infinity. In high dimensions, any delta will change it. Any delta will make it that I have the following behavior. If I again plot things versus time on these evolutionary time scales. Okay, so if I look at the psi, which is the fitness, so I'm going to look at the psi, how that behaves. This will come up, it'll come up, slow down, get slower and slower. Then it'll roughly saturate. Now, if this saturates, you can say, wait a minute, that means the evolution stopped. But no, it doesn't, because evolution is keeping going because the environment is changing, even though the overall fitness is staying the same. Okay. So the fitness, of course, now is not very well defined since the environment is changing, but this quantity which I've defined, the psi, is staying roughly the same. Okay. Now we can ask how far has it got? Okay, so we can draw a line up here. Okay, and this is the line of the typical maxima, the fitness at which I would find the maxima as a function of psi, a function of x. Okay, so if I fix the environment, I would find the find some maxima, and it's not getting to that. There's some gap there. So there's some gap as to where it gets to. There's a little gap gap here. Okay, there's a little gap there, and this gap is going to be proportional to some power of delta. Okay, the smaller delta is, the closer it gets, but it never quite gets to where the maxima are. So this is really going to be very much is the wandering around the saddles. Okay, how could I see that? How can I see that? Well, I could plot the index. So I can plot the index here. The index will come down and saturate. So this is now the index. So this means I'm getting closer to saddles. If this was 0, it'd be maxima. And this will saturate at a value here, which goes as some other power of delta. So it doesn't go to 0. So this is where I goes to infinity. Okay. If I'm more precise as to what I meant by over here, I meant that the psi will go to so psi at infinity will be approximately the psi at which the maxima, I first find maxima minus something which is delta the alpha. Okay, so I'm never seeing the maxima at all. I'm just keeping wandering around. And what this behavior is, it's deterministic chaos. So this behavior is that I get deterministic chaos. It's deterministic because I'm assuming no stochasticity. I'm just going uphill, but uphill in this weird way of uphill in this gradually changing environment. Okay? So of course, the effects of small delta, in the limit of small delta, this effects are small. It's going to get very close to this. It's going to slow down. So if I look at the typical size of the dx dt, if I look at the dx dt, typical magnitude of it with the evolution, this typical magnitude is going to go to 0, and it'll go as t goes to infinity. As t goes to infinity, this will go as delta to some other power gamma. Okay, and you can work out what those powers are in the concrete example, which I'm now going to show. Okay, so this is all very abstract. So but however, it's the result of at least semi-honest calculations. And so I want to at least say what the model is, which will give this. Okay. But the claim is that this is going to be very robust. There's a whole class of models that will give this behavior. Okay. So I'm now going to write down as a concrete model. Okay. For convenience, it's convenient to put the x's on a sphere, a hypersphere, with x squared, say equal to d. That gives you high symmetry, and you can do various analytical things. And a lot is known about such landscapes, random landscapes on spheres, but where it's a fixed landscape. Okay, so a lot is known about that, and all kinds of things about the statistics of the landscapes and the uphill dynamics on that landscape. The effects of fluctuations, temperature, or fluctuations like drift are known. Okay, so I'm going to now write down a particular model. I'm going to write down my psi. This is going to be a sum of all of the components. So my vector x has now components xi with I equals 1 to d, and this is some j I j k xi xj xk. So that's my landscape in the absence of the ecological feedback, and the j are IID Gaussian. They're independent Gaussian variables with some variance with mean 0. Okay, so that's my landscape. It's a simple form of a complex landscape and is known to be one of the generic classes of landscapes. But now I'm going to add a part to this. I'm going to add a part here. Okay. Plus a part here, which is now going to have my parameter delta in front of it. It's going to be small plus delta. And now I'm going to put again the sum on the I, j, and k of some other set of random variables, I, j, k. And this, of course, is going to depend on the x. That's the way it depends on the phenotype. Okay, but then it's going to have, instead of the x coming in there, it's going to have y k. Okay, so this is the way it depends on the environment. That's the way it depends on the environment. Okay, and my W's are also IID Gaussian. Okay, so I've got a Gaussian random potential on the sphere, the random landscape coming from this part, and then I'm putting a modification that comes from this, which depends on the environment. Okay, and then what is my dynamics? My dynamics is that dxi dt. I've got one component of it, and it is d by dxi psi. Okay, it's evaluated at y equals to x. Right, that's the environment that's there. It's now changing, coming from the mutations. Right, so this is the evolutionary dynamics on this landscape. So it's evaluated there, and then I need a piece which has a Lagrange multiplier to keep the this bit keeps it on the sphere. Technical term. Okay. Oops. Okay, so this is my dynamics, and then this is a model which one can analyze by methods I'm going to talk about tomorrow. So this is analyzed by methods for dynamical mean field theory. Damn. Oh, what the hell? Sorry about that. Okay, dynamical mean field theory, and I'm going to talk about how to do that tomorrow. It's analogous to things done for spin glasses, for those of you who are familiar with that, and I say I'm going to explain it tomorrow. Okay, so this is where the source of these predictions, the source of these understandings, and the one thing which we know from analysis of this is the kinds of behavior. So particularly the phase, this red queen phase, is generic. You can change the form. You can make this having quartic terms. You can put other things in here, and with small other changes won't change the behavior. And this is really then, in the sense of the physics, is like a generic phase. Okay. I should mention, why am I calling it red queen? It's red queen because you have to keep running very fast just to stay still. You have to keep up with the changes in the environment by changing, and that's what driving the evolution. And with delta small, you don't have to go very fast. It's not very red queen, but it's always have to keep racing and never really getting anywhere. So there's no sense in which is an overall improvement. Statistically, you can't tell the difference between whether you're at this time or whether you're at much longer times. Once you've got these initial transients, things will saturate, and it'll get to a statistical steady state out here at long time. Okay. Yes. So the question is: was the saturation of psi over long times on average? Well, this is one of the advantages of high dimensions. It is a bit like thermodynamics. Is the quantities will fluctuate, but the quantities will fluctuate by something which is much less than the amount in which they're averaged. So this will go to essentially a constant amount. It'll have fluctuations of relative magnitude, something like 1 over square root of d coming around this. It'll depend on delta as well. Okay, so it will saturate roughly. The index will come around. The index will also fluctuate around a little bit, but not by much. Okay, and this is the big advantage of the large dimensions, or one of the big advantages for doing it. The other is the ability to use these methods. Okay, so what I just want to, I'm going to explain how you do those kinds of problems a bit tomorrow, but what I want to do today is just ask some questions and extensions about this. Okay. So one question is there also a phase with different random potential without red queen, meaning that phi slows, everything slows down. It always just keeps slowing down. Everything just keeps slowing down. So that's one possibility. I think I know the answer to this, but I'm not sure. That's one possibility. Another one is if I'm in a simple environment, to the extent that exists, a simple environment. So there I would sort of think, well, in some sense, there's not going to be very many peaks. So there's a modest number of peaks, or at least fewer peaks. But I have a lot of evolutionary constraints on how y can change. In other words, it can only go in certain directions. Whether it can go in one direction or not will depend on where it's gone before. So that'll introduce what's like the saddles, and will introduce something which has a large number of possible places that it could get to, even though they're phenotypically sort of the same. There's fewer peaks in the organismic phenotype, but in the level of the nanophenotype here, the changes are constrained. Different changes could have similar effects. And so I could change in this way, or I could change in that way. It has similar effects. But a way, say, in between, this way, I'm changing like that, this would be forbidden by the constraints. Okay, so I'd have a large number of constraints. And so I would like to ask what happens if I have a large number of constraints. So there are many of these, and particularly actually I would like to ask about what happens if there are order D constraints. Okay, then one can analyze some things as well. And again, I have some sense of what happens, but not certainly don't fully understand. Okay. And the last one, and the most important one, is about diversification. Okay. In particular, I can get what's called evolutionary branching. Oh, and I should mention this whole framework. This whole framework is called adaptive dynamics. This framework of where the environment changes due to the evolution, and particularly Michael Dobley. Is it Dobley? Michael Dobley has an enormous amount of work on this, including finding chaos in some models of low dimensions. They're doing it numerically primarily, so can't do high dimensions. But one of the important phenomena that happens is you can get this evolutionary branching. So say I've got some, again, I've got some saddle in the here, and I'm coming along, and so I'm coming along here, and I'm coming along, say, getting near the saddle. Okay, but then I get a mutant. My mutant can go over to here. My mutant is now not infinitesimal. Okay, so this mutant then can go off in another direction. It comes here. It'll go off, say, another direction. Another direction. This will go this way, and these can coexist. So these two then can coexist. So now I've got two types. Okay, and the system will be, quite generically, will tend to be unstable to this branching. Meaning it'll be unstable to having coexisting types. Okay, so the question here is, if we ask, what is the question? Can you get large number of types, strains coexisting? Now, of course, some will go extinct. Other ones will be driven extinct deterministically. Other ones will branch. And so you could get a continuing turnover from this. And this is, I think, the most interesting question as to whether or not this can occur, because that starts bringing together the questions about the evolution and the ecology. So I don't know what the answer to this is. Possibly you could get maybe order D different strains. That would be, I think, one of the most interesting possibilities. This I don't know. I've some thoughts on it, but not very advanced. Okay, this I'm going to talk about now tomorrow. This I'm going to talk about tomorrow in the general question of many strains coexisting, not in this framework, but in back the Volterra one, and then come back to this sort of question at the end of on Friday. Okay, so the main messages here is that evolution in high dimensions is very different than in low dimensions. Okay, it's very different than one's intuition. In particular, that any small amount of ecological feedback, any small amount of that will make the evolution continue forever. So in some sense, if evolution does continue forever, even in a simple environment, we shouldn't be too surprised. Okay. So I'm going to say stop there and take questions. And sorry, I suspect I've gone on a bit longer than I should have already.
So
questions.
we're waiting for questions.
Yeah, if you want to unmute and ask the questions directly.
Can I make a comment? Hello.
Yes.
Hello, Daniel. Thank you very much.
Hi, Mercedes. Hi.
Very interesting to try to understand you. I think there is a fascinating question, which is when you get this interaction of ecology and evolution, how does the same evolutionary forces that have to do with coexistence of the organismal phenotypes will determine the dimensions of the nanophenotype? So how big is the variation, the pool of variation from which you assemble the high coexistence at the higher level? I think those things are connected. And the way they are connected may be super important for thresholds that we may cross when we set up conditions where we lose the capacity of keeping a diverse nanophenotype. So I think this is a fascinating question. I think where you have hyperdiverse systems in biology, evolution over long times and large space has set up a very high dimensional nanophenotype. But that's not by chance. And I think understanding that connection is critical to maintaining diversity.
So that's a great point, or I guess many great points in there. So most of the interesting properties of the environment that an organism fields are properties made by the rest of the biology. And so they say the simple example, when you take a cell, which is complex, you lyse it, you've got all those chemicals in it, all kinds of potential. Of course, with geographical structure, the biology will also set up gradients, set up different conditions, and you'll get the development of all the further complexities which enable the more evolution and more diversification. Okay? So the way I want to sort of try to ask that is that something which is sort of, we should expect that once the biology's got complicated enough, okay, much less complicated than it is now, once it's got complicated enough, do we generally expect that one will get in this state or this phase of continuing diversification, the environment in some sense continue to get complex as well? But even if we sort of saturate how complex the environment is, bacteria are not that much more complex now than they were 1 billion years ago, and we can still have everything continuing once we get to such a state. Okay? But there's a question, as you say, about sort of threshold, as so some threshold one has to go over where this sort of picture might start to apply. Okay. Yeah, in particular, there's in some of the parameters which I had here. Of course, D isn't infinite; it's large. For any finite D, there's a delta, but if it's not big enough, if you don't give enough feedback, then things will just slow down and you'll go to a maximum. Okay, so you sort of need the D to be big enough to get this going, and then the question is, once it gets going, does it sort of continue? And should we be surprised that it continues? I'm going to say a little bit tomorrow about thresholds for diversification in Lotka-Volterra models in absolute simplest situation. Again, I don't know whether any of this has anything directly to do with the biology. It's really the goal is to sort of make some conceptual things and give them some sort of mathematical footing in the sense of knowing this is something which could happen with sufficient complexity. But a really interesting question, and to some extent, maybe I make some comments on this Friday, is that one can ask if you start with something which is not very complex and get the complexities added by as the evolution occurs, can you sort of get into some kind of high diverse state?
Yeah, I'll give an example tomorrow, perhaps in the malaria case, under very high transmission of the existence of such threshold. It may be of interest.
Ah, great. Well, so I was going to make a point. I listened to your lecture yesterday and really enjoyed it. And I was going to make a point of listening to it tomorrow. And one of the advantages of doing things on whiteboard rather than slides is I can modify in real time things based on the other.
Just to know that there is a connection, and I'd be interested in that connection.
Yeah. So I think these are good questions, I guess, for the roundtable next week also. So
yes.
Are there some questions from students or postdocs? Yes. Okay, from Roberto, is it physically reasonable to think about a rotating saddle environment like the electric field in a pull trap for ions, so that X is trapped in periodic dynamics. Okay, so that's interesting. So one of the things that Michael Dobley and collaborators find is that you can get things in these kinds of conditions where you get, you get a limit cycle, you get some rotating around. As it's going around, it changes the environment and it just goes on a cycle. You can also get where it branches. You can get where it branches and you have two of them going on, both going on cycles. So something like this would go on a cycle, this would go on a cycle, and they'd go around and do that together. So you definitely can get cycles. Generally, and I'll say something about this tomorrow, generally in high dimensions, cycles tend to be sort of unstable. You generally tend to have either sort of static things or chaos. Okay, and so in simple situations, you can get cycles, but you probably, if you go for a while, one of the organisms will find a way to sort of get out of that by doing something different, and it'll go off in that direction. Other questions?
So when you show the you have these plots of the index saturating, the index of the fixed point saturating at some low value. So are these saturations usually like, do they happen fast, or is there some time scale, like an exponential?
Well, okay, so there's a time scale. So there's a time scale associated with this. Okay, so there's a time scale associated with this coming down and this going up, depending on initial conditions. So if you thought of going into a new environment, externally controlled environment, then initially wouldn't be very fit. You would go up here. This would be pretty much independent of delta. It's small changes. You're just doing better in that environment. But these small ecological changes, and they would only really start to matter when you'd sort of got near to what would be maxima in that environment that wasn't changing. Okay. So there's a time scale associated with this, which is just sort of basic time scales in the model. When you go out over here, you don't converge this exponentially. You converge this as a power law. So you converge this power law convergence towards these at this end, and so you get slow convergence. And that's the case also if you look at the slowing down here. There's sort of a power law slowing down rather than exponential slowing down. Okay, but there are characteristic scales. I've sort of set them all to one to make life simpler.
So does that imply that if you could, so the rate at which new mutations fix would also go down as a power law, I guess? Is that?
Yeah. So this is the rate of which this is in fact I should have written it down. This is the rate of mutations fixing. Okay, so that'll go down, and how big the effects of those mutations are, so how fast they fix, will also go down, but it won't continue to go down, right? As you go towards for infinite times, this will still this won't slow down completely. It'll keep going at some rate, depends on delta. If we turned off the ecological feedback, it would just get slower and slower.
Okay, thank you.
So maybe I have a question. Hi, Daniel. I'm sorry I lost the beginning of your lecture, so probably you already addressed this, but so the question is, this is based on deterministic picture of the dynamics, if I understand. Right. So are you thinking about, and the question is, what is the effect of a noise on this? So are you thinking your species, I mean, a quasi-species picture where essentially your variables are robust with respect to noise, or so how does?
So for now, today, I'm not talking about quasi-species, because at any given time I have one strain, and then a mutant comes in and the mutant can take over. So I have two strains temporarily. So I only have one strain at a time. Okay. Certainly, the fluctuations will be demographic fluctuations. There'll be fluctuations in the mutations, and particularly this thing I showed here about the evolutionary branching. This mutation actually has to occur some distance away. It can't be infinitesimal. Otherwise, you don't get this. So it occurs some small distance away. So that's a stochasticity associated with the mutation. So any stochasticity will just add to it looking more chaotic, right? If it's deterministically chaotic, then a bit of stochasticity just sort of adds to that. If it's not chaotic, So if I'm in a situation like I get in 2 dimensions or in low dimensions, where I have a small number of small numbers of maxima, and I go, I'll just go to those and determine by the saddles. Then when I get near a saddle, of course, the fluctuations will be important. And so which direction I go here will depend on the mutations. Okay. So there's an analogy with about evolution, which I give when giving talks to physics audience or general ones. Which is the analogy is of a qualifying exam in physics, in which some departments, it's traditional to ask one general question, or sometimes even a thesis defense. And the professor asks the student, how do you measure the height of a building with a barometer? So the student answered, I throw it off the roof and measure how long it takes to get to the ground. So he was failed on that part, but the professor realized that was kind of unfair. So he asked one of his colleagues to re-examine the student. And she did, and she wrote the story down. So it's actually based on a true story. I'm sure it's been embellished. So she asked him, well, what other way would you do it? So he said, I would go up the stairs and use it as a ruler and measure the height of the building. She goes, what if you didn't have access to the building? She goes, well, then I would measure the shadow of the barometer and the shadow of the building, and I would get the height from that. So she said, what about something that uses more interesting physics? So he said, I would make a very good pendulum with the barometer, and I would measure the period of the pendulum on the ground, the period of pendulum on the top of the building, and being able to get its height from the change in gravity from the bottom to the top. She goes, what about something that uses its value as a barometer? So he says, well, I would go to the superintendent of the building, and I would say to him, if I give you this really nice barometer, I'll give you this very nice barometer if you tell me how tall the building is. So at that point, the professor said, you pass. And as they were leaving the room, she goes, but surely you know what the right answer is. And he says, well, I know what answer you want, but I see no reason that I should give it. And I would say all of the lessons from evolution experiments in the lab is that you try to select on one thing and the organisms do something different. The very first chemostat experiments, Leo Szilard, or early ones, where he was trying to select on faster growth rate, the bacteria were getting all flushed out. What did they do? They evolved to stick to the walls instead. They just played a different game. Okay, so this enormous number of possibilities, and I think this goes back to Mercedes' question, is if you think about the number of sort of possible ways in which organisms can do better in some conditions, that ways gets more and more, the more complicated the biology and the ecology, meaning how many other species and things are around, gets. Okay, so there's really there you get into the more and more possibilities. The stochasticity will certainly be important. It'll change things in detail, but in big populations, the stochasticity doesn't have to change things intrinsically. Okay, and I think that's related a bit to what I'll talk about tomorrow. We'll mostly talk about deterministic things, so we understand a little bit about putting in stochastic effects. So I'm a believer that for microbial populations, except possibly pathogens, in some extent drift doesn't matter. Extinctions matter. Drift matters when you have only one of you, but it doesn't. Otherwise, the effects of the drift are not very important. The stochasticity, as far as mutations, is certainly important. But even that, there are some aspects of it which don't have to be, such as say the statement that I got once. You're over here, you will tend to go there. You're not so likely to jump all the way over to here, even though of course, small mutations can have big effects. I don't think that really answered your question, but that was.
Well, yes. Thanks.
So I said tomorrow I'm going to be much more concrete and deal and work through things with concrete models and so on, and be more precise about what the kind of questions and things that I ended with here.
We have just one more question.
Yeah, some kind of traveling wave effects. So there's very interesting things, and this I will say something about as to what happens if you put in spatial structure. And so by spatial structure, I'm going to mean the simplest possible. The external environment is the same everywhere. Things can move around or get carried around, and ask what the effects of that are. Okay, and there are very big effects of that. In some circumstances, they can sort of look like wave effects. But when you get lots of traveling waves, they tend to again get more chaotic, like in the ocean. And so there are again sort of chaotic things can dominate. But the spatial traveling waves are certainly important. Waves in phenotype space. That's like a question that was asked previously about sort of cycles in the phenotype space. So all kinds of things are possible. which things are sort of generic in the sense of not special to particular models, that one has to get at. And that's really, sort of deep methods developed in physics in the last 50 years enable one to sort of think about that, to talk about which things are more generic. And again, I'm going to do example tomorrow with Lotka Volterra of some very interesting behavior that's completely non-generic, but I will use that to get some more understanding of some more generic, more robust behavior that shouldn't depend on all of the details. A question: I imagine evolutionary branching in 2 or 3 dimensions as a possible spherical wave. I'm not sure I understand that. I think here there's a discrete number of strains at any time, and so it isn't that there's a whole sphere of them branching out. It would be two points that would be separating, and then of course one of them could branch again, and you can get more. A radiation going in a new environment, completely new environment. You can get a lot of radiation into different into many different directions, all happening simultaneously, and of course the mutations can be fast enough and things that you'll get a lot going on at once.
Okay, so it appears that we don't have any further question. Thanks very much, Daniel, for the beautiful.
And if you have further questions or you want clarifications of things that come up overnight, then please email me and I'll try to address those. Probably generally tomorrow. Thank you.
Thank you very much, and see you all tomorrow for a new session. Goodbye.
Bye.
Hola, Armand. Ciao.
Oh, perdón. I just saw that. I just saw your message.
We are still there, so I don't know why you appear in my screen. People are still there. I'm leaving right now.
Okay.
I'll see you sometime.
Well,
Ciao.
do you want to?
Thank you.
Ciao.
No, we should not talk to this channel.